we are talking about the process of gametogenesis, and what we find is we are beginning with what is called as the zygote. Zygote is going to have one maternal as well as paternal nucleus, and each of the nucleus is bringing haploid number of chromosome. So the zygote will be 2n and 2n, where this small n represents the number of chromosome, whereas the capital N is the amount of DNA. And it is understood that the maternal nucleus will bring 1n, whereas the same will be brought by the paternal nucleus, which is again 1n and 1n. Though there are total 46 chromosomes, we are representing them all with the 2n and 2n. 23 pair of chromosomes, where one set of chromosome is contributed by mother and the other set of chromosome is contributed by the father. This is just a way of representing with a formula called 2n, 2n. Now this zygote is continuously dividing and forming multiple cells. What we get later is called as the blastocyst. And in the blastocyst we are having the inner cell mass. The inner cell mass is having what is called as the epiblast cells, and it is the epiblast cells which are going to form what is called as primordial germ cell. Now primordial germ cell, which is formed by the epiblast cells, is the first sex cell of our body, which is then going to give us the gonocytes, the gametogonia, the primary and secondary gametocytes. It is understood that the first sex cell of our body, that is the primordial germ cell, is capable of forming the male as well as female sex cells depending upon the presence or absence of Y chromosome. If we have the Y chromosome, then we'll have the formation of sperms by the process of spermatogenesis. But if we do not have the Y chromosome, then the same primordial germ cell is going to form the oocyte by the process of oogenesis. And later we know the mother gives the oocyte, the father gives the sperm, bringing 1 unit of DNA with themselves and 23 pair of chromosomes, one set from mother, one set from father, and hence finally we are getting a single cell which is the zygote having 2n, 2n genetic designation. Now in this process we are having some cell divisions involved, that is the mitosis and meiosis. Let us look at a diagram to differentiate between mitosis and meiosis. We need to remember that mitosis is one equational division, means it is not going to change the chromosome number, whereas the meiosis is a reduction division, especially when we are talking about meiosis 1. We should understand that the meiosis is two-stage process, where you will find that in the beginning it is meiosis 1, which is going to reduce the chromosome number to half. That is why it is called reduction division, whereas meiosis 2 is just like mitosis. It does not change the chromosome number. As you can notice, we are starting with a cell which is a normal cell having 46 chromosomes and designated by the term, as we have mentioned earlier, 2n and 2n, where the small n is the number of chromosomes and the capital N is the amount of DNA. So we get 1/2 of chromosome pair from maternal side and the other half from the paternal side, each parent contributing to 1 unit of DNA. Though there are total 46 chromosomes, but here we are designating only one of them for the sake of simplicity. And during mitosis, which is equational division, the initial genetic designation does not change in the daughter cell. You will see that even the daughter cell are having the same designation. So they are again diploid with 2 units of DNA. Now, how is it happening? Let us look at the detail. We are going to zoom here and we understand that before any cell division, there is a resting phase, the interphase, and there also in the S phase, synthetic phase of interphase, there will be DNA duplication. Before any cell divide, it must duplicate its DNA, and that is why this 2n 2n status will change into 2n 4n. We'll see that there are sister chromatids available and each sister chromatid has 1 unit of DNA, so total 4 units of DNA, though the number of chromosome is still diploid. So here you can mention before it enters the cell division there is DNA duplication, and now the designation is 2n 4n. Further in mitosis we'll have prophase, metaphase, anaphase, telophase, and it is during the anaphase we find that the chromosomes are aligned at the equator and separating. What we notice here is the maternal and paternal chromosome having the sister chromatids at the centromere will be breaking, and one of the sister chromatid from maternal side and one sister chromatid from the paternal side will move to the opposite poles. And finally what we get is 2 daughter cells, but having again a diploid status with 2 units of DNA. As we have mentioned, in mitosis there is no change of chromosome number or the amount of DNA, but it does not happen happen in similar fashion in meiosis. As we know, in meiosis there will be haploid cell with 1 unit of DNA. These are the gametes, and the gametes must be haploid with 1 unit of DNA. Then only the union of male and female gamete will restore the normal genetic constitution, which is 2n and 2n. Let us focus upon meiosis as well now. As we have mentioned, meiosis is a reduction division, especially the meiosis 1. Here you will see this is going to be meiosis 1, and beyond that is meiosis 2. But meiosis 2 is just like mitosis. And before any cell division, we must have the DNA duplication. So focusing upon the meiosis, in the resting phase, which is interphase, S phase of interphase, there will be DNA duplication. And as we zoom here, synthetic phase of the interphase changes the normal 2n, 2n status of the cell into 2n and 4n. So you can see there is duplication of DNA. Now in meiosis 1, there will be prophase 1, metaphase 1, anaphase 1, telophase 1. And in the prophase 1 itself, it will have 4 subdivisions. So in the prophase 1 of the meiosis 1, we'll have the leptotene, zygotene, pachytene, and diploatine. And it is in the pachytene, we remember one mnemonic, PTC. P represents the pachytene and T is the tetrad. Means you have the 4 counting available because there are 4 sister chromatids. You can count them 1, 2, 3, 4. So it is a tetrad and involved in what is called SC, the chiasma. At the point of chiasma, you'll find there is some exchange of genetic material between maternal and paternal chromosome. And finally, what you get is the paternal chromosome having a piece of maternal genes and the maternal chromosome having a piece of the paternal genes. So genetic recombination during the chiasma formation is also identifying meiosis from mitosis. So this genetic recombination is a distinguishing feature of meiosis. P for the pachytene, T for the tetrad, and C for the chiasma. Now during meiosis 1, we see the maternal and paternal chromosomes are separating. That is why the final cell which we get is a haploid cell with 2 units of DNA. As you notice here, we can zoom. After meiosis 1, we are having the primary gametocytes, which are haploid with 2 units of DNA each. Now, this is the primary gametocyte, which could be either male or female. Mean to say this could be a primary oocyte or primary spermatocyte. In meiosis 1, the maternal and paternal chromosomes separate, and each cell is a haploid cell. Then, in meiosis 2, which is just like mitosis, there is no change of chromosome number. What happens is just the sister chromatids are going to separate, and that is why the final cell, the gamete, is having haploid status with 1 unit of DNA, that is N and N. And one of the gamete from maternal side, the other gamete from paternal side, as they undergo fertilization, the normal status of a cell is restored 2N and 2N. Now let us focus upon the details of oogenesis in another diagram as well. Here we are focusing upon the female gametogenesis, that is the oogenesis, though the process will be same for a male as well, that is the spermatogenesis. There are some differences, like in a female, that is oogenesis, there will be some polar bodies formed, actually 3 polar bodies, and only 1 gamete is coming as a resultant product. Though in male it is 1:4 ratio, there are no polar bodies and will actually have 4 gametes which are capable of fertilization. But for a female, only 1 cell is capable of getting fertilized, and it is the secondary oocyte. Now, as we understand, the primordial germ cell is the first sex cell of our body and is going to form the gametogonium. In this case, it is a female, so oogonium, which will then form the primary gametocyte, the primary oocyte. During this process, what we get is mitosis involved. So, from this stage to this stage, it is the mitosis going on. But beyond that begins the meiosis, and that is meiosis 1. As you can see here, the first meiotic cell division. The cell which enters the first meiosis is the primary gametocyte. In a female, it is primary oocyte. And we have to remember, primary gametocyte is the largest of all the cells in the process of gametogenesis. Then we have to remember one more thing. For a female, the primary oocyte is arrested in prophase 1, that too in the diploatine stage till puberty. And we remember a mnemonic P1P. Now, what is this P1P? It says that the first P, the primary oocyte, is arrested in prophase 1, the second P1, and the third P is puberty, because only after puberty there will be LH surge and the primary oocyte can become the secondary oocyte. For the first meiosis to complete, you must have LH surge. So you can mention only after LH surge will have the release of first polar body and the secondary oocyte. As we write down here, LH surge is essential for the completion of the meiosis 1, and after that only we'll have the secondary oocyte and the first polar body. Then it is the secondary oocyte which will undergo ovulation and fertilization. Only after fertilization, the second meiotic division will be completed. So it is the secondary oocyte which is undergoing the process of ovulation followed by the fertilization, and after fertilization only we'll find there is formation of the second polar body. So second polar body is formed after fertilization. What about the first polar body? As we can see, second polar body is seen only after fertilization, whereas if we are talking about the first polar body, it is formed after LH surge but before ovulation, which you can mention here. The first polar body is formed after LH surge but before ovulation. We will mention this fact again in another diagram. These are all MCQs which come frequently, and we should have specific information to answer them. Here we are plotting the concentration of LH hormone variation during the menstrual cycle. Normally, menstrual cycle is taken as 28-day cycle. Day number 1 is the first day of the last menstrual period, and then total 28-day cycle. It is the day number 14, that is the mid-cycle, will have the process of ovulation. Now, as we are plotting the concentration of LH hormone, there is a sudden rise of the LH hormone concentration just before ovulation. That is called LH surge, and is happening approximately 36 hours before ovulation. We can mention it here. The LH surge is happening approximately 36 hours before ovulation. Then what about the LH peak? The maximum concentration of the LH hormone, that is approximately 12 hours before the mid-cycle, the day of ovulation. So you can mention that here. The LH peak is happening about 12 hours before ovulation, and that is the time when the first polar body is released. So first polar body is released approximately 12 hours before ovulation. When is the second polar body released? Second polar body is released after fertilization. Then when is the fertilization happening? As we mentioned, the polar body number 2 is released after fertilization. We have to mention the fertilization should occur within 24 hours of the ovulation. So the window period a female is having after ovulation is just 1 day, 24 hours. If she is going to have a baby, the baby or the zygote must be available on day number 15. So as you can see, the window period for a female is just 1 day, and day number 15 she must have the zygote. Otherwise, the secondary oocyte is degenerated and is menstruated by the end of the cycle, and she has to wait for the next menstrual cycle to have fertilization possible. Now, what about a male? Once the sperms are ejaculated in female, the sperms are capable of fertilization. For how long is a question. So once the sperms are ejaculated in female, they are actually having 2 days or 48 hours when they are capable of fertilization. And what about the survival time? Once the sperms are ejaculated in a female, how long they can survive in the female genital tract is another question. For that, you can mention the survival is possible for about 5 to 10 days, though they will not be capable of fertilization. Maybe sometime it is rare. It has been observed fertilization will be happening on 7th day after ejaculation, but that is not normal. Normally, they have the capacity for fertilization only for 2 days after ejaculation, and we are not supposed to discuss the rare events. That long days of survival is of no use. It means that if the sperms are ejaculated on day number 12, 13, then there is a chance of fertilization. As you can mention here, sperm ejaculated on day number 12 or 13 or 14, then we will have the fertilization happening. Not if it is before day number 12 or after day number 14. So sperms ejaculated before day number 12, no fertilization possible. Or after day number 14, no fertilization possible. The most fertile 3 days are around the day of ovulation. That is day number 12, 13 and 14. So this is the most fertile period. Male having the 48 hours after ejaculation and female having only 24 hours after ovulation. The most fertile 3 days are around the day of ovulation. And the next diagram is going to talk about the process of ovulation, fertilization and implantation as well. This diagram shows us the uterus where the implantation will be taking place. But before that, we see that it is the ovary which is undergoing ovulation, giving us the secondary oocyte, which is then undergoing fertilization in the ampulla of the fallopian tube. So it is the ovary which is having the Graafian follicle is giving us the secondary oocyte. This secondary oocyte is covered by a glycoprotein membrane which is called zona pellucida to attract the sperm for fertilization. And the fertilization should occur within 24 hours of the ovulation. Once a single sperm nucleus is inside the secondary oocyte, the zona pellucida undergoes zona reaction, deflecting the remaining sperms, preventing polyspermy. Zona pellucida is also preventing abnormal implantation. Now, day number 1, we have the single cell structure which is called zygote. Day number 2 will have 2 cell stage, and day number 3 will have the morula. The morula can have 12 cell stage, or it could be 16 cell, 32 cell, 54 cell. But more specifically, if a question comes, we should answer it as 16 cell stage on day number 3. Then there is one advanced morula on day number 4 entering the uterine cavity. And this advanced morula can have any number of cells. Could be 32 cell or even more than that. Then on day number 5, it will change into a structure having some blast cell and a cyst-like cavity. We are calling it as the blastocyst. But this blastocyst is still covered by zona pellucida, which is preventing implantation. It's on day number 5, the blastocyst will be hatching out of zona pellucida and then attaching to the uterine endometrium. And implantation is a week-long process beginning on day number 5, completing on day number 12. Post-ovulation, or you can say post-fertilization, because the day of ovulation is also the day of fertilization. Fertilization should occur within 24 hours of the ovulation. Now a question comes, when does the implantation happen? You say day number 5 to 12 post-ovulation or post-fertilization. But if that option is not available, more precisely you can say day number 7 to 9. So it is day number 7 to 9. More precisely, the process of implantation is taking place in the endometrium. Let us revise this process of entry of the morula and its further development in the blastocyst stage in the next sequential diagrams. We are magnifying the details which are happening inside the uterine cavity here. Like we see, this is the advanced morula which has entered the uterine cavity on day number 4, and it will be having cell number which could be 16 to 64. And then it is going to change into what is called as the blastocyst. So when we are talking about the blastocyst, it will have some blast cells and a cyst-like cavity. A magnified picture is shown here. This is the uterine endometrium and this is the blastocyst having some blast cell and cyst-like cavity. There are 2 types of blast cells. The inner cell mass is the embryoblast going to form the embryo, whereas the outer cell mass is the trophoblast. Tropho means nutrition, and they are going to contribute towards the placenta, providing the nutrition. So we can mention here the inner cell mass is the embryoblast going to form the embryo, whereas the outer cell mass is the trophoblast. Tropho means nutrition. They'll contribute to the placenta formation. Now they themselves will be dividing into 2 types. The trophoblast will become cytotrophoblast and syncytotrophoblast, and the embryoblast will become epiblast and the hypoblast. And this will be happening on day number 6 to 8. That is the transition of first week into the second week. Like here you can see some of the trophoblasts, they have lost their cell margin, becoming syncytium, hence called syncytotrophoblast, and the cells which have intact cell margin are called cytotrophoblast. It is the syncytotrophoblast which will help us in implantation of the conceptus into uterine endometrium. The week number 2 is called as the week of 2, as we have 2 type of trophoblast cell, 2 type of the embryoblast cell, and 2 cavities as well. So here, as you can see, this is the diagram which we have magnified here, and we can still magnify it further. And as we do that, on the dorsal side we have got the amniotic cavity, and on the ventral side we have got the yolk sac cavity. So here, this is the dorsal side where the conceptus is getting into endometrium, and on the dorsal side we have the amniotic cavity, whereas on the ventral side we have the yolk sac cavity. And the embryo is in the form of disc. The dorsal cell is called as the epiblast cell, and the ventral cell is is the hypoblast cell. Both of them have come from the embryoblast or the inner cell mass. So the epiblast cells at the floor of amniotic cavity, whereas hypoblast cells are actually at the roof of the yolk sac cavity. They are more ventral comparatively. And in fact, the hypoblast cells, they'll be removed from the embryo, and the cells which contribute to the embryo are only the epiblast cells. So week of 2, we have 2 type of embryoblast, 2 cavities. What about the 2 type of trophoblast? The cells which are having intact cell margin are called cytotrophoblast, whereas the cells which have lost their cell margin is syncytial trophoblast. And here is the formation of the villi. There is a primary villus, secondary villus, tertiary villus. In the primary villus, we'll have 2 type of trophoblast cells. Towards the maternal endometrium, the syncytial trophoblast. Towards the embryo, the cytotrophoblast. Together, they will form the primary villus. Later, they will change into secondary and tertiary villus. We'll see that in the next coming diagrams. But at the moment, let us move from the second week to the third week of development. In the third week of development, there will be formation of 3 germ layers. Process is called gastrulation, and the 3 germ layers are formed from a single cell, which is called as the epiblast cell. Epiblast cells will be undergoing proliferation to form primitive streak, and then they start migrating towards the ventral side, and which can be seen in the next diagram. And as you can notice, it is the epiblast cells which are proliferating to form a structure which is called as the primitive streak, and then they start migrating ventral towards the yolk sac and replacing the hypoblast cells, taking their place and forming the first germ layer, which is called as the endoderm. So as you can notice, this is the primitive streak, which is proliferation of the epiblast cells, which are now moving down ventral. The process is called ingression of epiblast cell and replacing the hypoblast cell, taking their place to form the first germ layer, which is endoderm. Now, endoderm is the first germ layer to form, then forms the next layer, the mesoderm, and lastly, the epiblast cells on the dorsal side will change and become the ectoderm. So, the sequence of formation of germ layer is endoderm, after that it is the mesoderm, lastly it is the ectoderm. But all the 3 germ layers coming from the epiblast. In the third week, by the process of gastrulation. Let us look at this process in another format as well. As we understand, the dorsal cavity is the amniotic cavity and the ventral is the yolk sac cavity in the second week of development. And then we have got epiblast cells at the floor of amniotic cavity towards the dorsal side and hypoblast cells at the roof of yolk sac cavity comparatively to the ventral side. But in the third week of development, the epiblast cells, they are proliferating to form the primitive streak. So this primitive streak is formed by the proliferation of epiblast cells, which are all shown here. And this primitive streak formation is in chordocephalic direction. It begins towards the tail of the embryo, that is the caudal end, and is migrating towards the head end or cephalic end of the baby. So as is evident, the primitive streaks start forming at the caudal end, the proliferation of epiblast cell, and migrate towards the cephalic end or the head end. But the process of gastrulation is exactly the opposite. Epiblast cell will be forming the 3 germ layers, first towards the head and then towards the tail. So formation of primitive streak is chordocephalic, whereas gastrulation is cephalocaudal direction, exactly opposite direction. That comes as a question. Now, these epiblast cells, which you are looking at the dorsal view of the embryo, they are migrating ventral, the ingression of epiblast cells, and through the primitive streak, they are going ventral. As you take a section, you can find here, this is the amniotic cavity. At the floor, we have the epiblast cells migrating towards the ventral side at the roof of the yolk sac cavity, replacing the hypoblast cells and themselves becoming the endoderm. So first germ layer to form is endoderm at the roof of the yolk sac cavity. Second layer to form is the mesoderm, and third layer from the epiblast is the ectoderm, which is at the floor of the amniotic cavity. You can draw the 2 cavities once again. This is going to be the amniotic cavity on the dorsal side. This is the yolk sac cavity on the ventral side, and formation of the 3 germ layers in a sequence: endoderm, mesoderm, and ectoderm, all from the epiblast cell. So what you see here on the dorsal side will be ectoderm. If you remove the ectodermal layer, then you'll have the mesoderm. And if you still remove the mesodermal layer, then you will have the endoderm layer. As you can show here, this embryonic disc is going to have 3 layers in a sequence. The most ventral will be the endoderm, first germ layer to form. Sandwich in between will the mesoderm, and most dorsal is now epiblast cells changing into the ectoderm. In the next diagram, let us look at the consecutive stage of this structure which is a transverse section of the embryo after gastrulation. And as we see, there's a dorsal cavity, amniotic cavity, and ventral is the yolk sac cavity. After gastrulation, we have the three germ layers. Ectoderm at the floor of the amniotic cavity, which is the dorsal most. Sandwich between is the mesoderm, and most ventral is the endoderm, which is lining the yolk sac cavity. This notochord is the axial mesoderm, which is important because it is going to stimulate the overlying ectoderm to form the nervous system. The ectoderm will be two types. In the central portion, we have the neuroectoderm overlying the notochord, whereas in the periphery we'll have the surface ectoderm. The neuroectoderm forming the nervous system and surface ectoderm forming the structures like the skin, epithelium. So neural tube will form from the neuroectoderm. Mesoderm will form some tubes like cardiovascular tube, the heart and blood vessels. Whereas the endoderm, which is lining the yolk sac, is going to form the gut tube. We'll be talking about them once again, but let us just revise the formation of primitive streak once again. This is the dorsal view of the embryonic disc. We are in the amniotic cavity, looking on the dorsal view of the embryo, which will be all epiblast cells. They are proliferating to form the primitive streak. As we mentioned, primitive streaks start forming from the tail end, caudal end, and moves towards the head end or cephalic end. Towards the cephalic end, there will be formation of the buccopharyngeal membrane, and towards the tail end, there is a formation of the cloacal membrane. The primitive streak itself will have three components. As you can see, there will be a primitive groove, a primitive pore, and a primitive node. And we are going to draw this ourselves as well. Looking at the dorsal aspect of the developing baby, taking this as the caudal or the tail end, and to that side is the cephalic or the head end, we draw the proliferation of the epiblast cell forming the primitive streak, having three components, which we name as the primitive groove. And then we have the primitive node and a pit, which is the primitive pit, also called as the blastopore. So this is the primitive streak developing in caudocephalic direction, having a groove and a node and a pit or the blastopore. On the dorsum, there are a lot of epiblast cells which you can show, and they are the ones which were proliferating to form this primitive streak. Now, these epiblast cells, which we are telling here, contributing to primitive streak, they are now migrating ventral. The process is called ingression. And as they migrate, they jump into this pit and then they are going ventral towards the yolk sac cavity on the ventral side and then form the first germ layer, which is the endoderm. So we can mention the first germ layer on the ventral side to form is the endoderm. And then the same epiblast cell will form a layer sandwiched called as the mesoderm. And later, the epiblast cells which never migrate, they remain dorsal, will be contributing to the ectoderm. So all the three germ layers, they are all coming from the epiblast, migrating in the primitive streak. Now, towards the cephalic end, we find there is a buccopharyngeal membrane having no mesoderm because the superficial ectoderm fuses with the deeper endoderm with no intervening mesoderm. Same thing will happen towards the tail end, the cloacal membrane. It has no mesoderm because there is a direct fusion of the dorsal ectoderm and the ventral endoderm. You can mark it here. The cloacal membrane towards the tail end and the buccopharyngeal membrane towards the cephalic end, they are having no intervening mesoderm because it is the direct fusion of the overlying ectoderm and the ventral endoderm with no intervening mesoderm. But there is some mesoderm between the primitive pit and the buccopharyngeal membrane. Some of the epiblast cells, they ingress here and move towards the buccopharyngeal membrane to form what is called as the axial mesoderm or the notochord. So let us show the migration of the epiblast cells, which then lie between the primitive pit and the buccopharyngeal membrane to form the notochord. And the epiblast cells which are forming this notochord is called as the axial mesoderm. This notochord is important to stimulate the overlying ectoderm in the central portion to form the nervous system, whereas the ectoderm towards the periphery is surface ectoderm. Like we said, it contributes to skin, epithelium. Here, if you remove the ectoderm, deep to that will be the mesoderm. And if you remove the mesoderm, then deep to that will be the endoderm. That is the orientation. Then we continue with the transverse section of the embryo showing the three germ layers and discussing that the ectoderm form the neural tube, the mesoderm form the cardiovascular tube, and the endoderm contributes to the gut tube. Let us take the transverse section of the embryo after gastrulation then and discuss the formation of the three tubes. We already have seen this diagram where we were talking about the notochord stimulating the overlying ectoderm, which will then form the neural tube. So here we can mark the neural tube and mention that this neural tube is actually coming from the neuroectoderm, which is central in portion, forming a neural plate, forming a neural groove, and then neural groove is going to become a neural tube. So as you can see, there's a neural plate becoming a neural groove, and this neural groove is then becoming a neural tube to form the central nervous system, that is the brain and spinal cord. Then what about the gut tube? Where is the gut tube coming from? The gut tube is actually coming from the endoderm lining the yolk sac cavity. As you will notice, we can mention it is the endoderm lining the yolk sac cavity which is going to form the gut tube here. So this is the gut tube which is coming from the endoderm lining the yolk sac cavity. Now once you have mentioned about the endoderm lining the yolk sac cavity contributing to the gut tube, what about the cardiovascular tube, which will have cardiac muscles, smooth muscles, the heart and blood vessels? That cardiovascular tube is coming from the mesoderm, that is the middle layer. Sandwiched between the dorsal ectoderm and ventral endoderm is the mesoderm. It is this mesoderm which is contributing to the cardiovascular tube. As you can mention, sandwiched between ectoderm and endoderm, this mesoderm is contributing to the cardiovascular tube. Now, once we have discussed that cardiovascular tube or the heart tube, blood vessels, they are coming from the mesoderm. There are 3 germ layers and there are 3 cavities also forming. We already know amniotic cavity on the dorsal side, yolk sac cavity on the ventral side. There's a 3rd cavity developing which is called intraembryonic coelomic cavity, and which is all around the baby. So you can see it on the right side as well as left side. Dorsal amniotic cavity, ventral yolk sac cavity, and on the sides you have coelomic cavity. So that coelomic cavity which you find here developing laterally on either side later forms 3 more cavities. That is the pericardial cavity, the pleural cavity, and the peritoneal cavity. And their membranes like pericardium, the pleura, and the peritoneum, they'll be coming from the mesoderm lining the intraembryonic coelom. Because of that intraembryonic coelom, the mesoderm will be dividing into 2 parts, and they are contributing to parietal and visceral layers of the pericardium, pleura, and peritoneum, which we'll be discussing in the later diagrams. You can see if it is the heart tube, the cavity formed is the pericardial cavity. If it was the lungs, then it will be pleural cavity, and if it was the gut tube, related to gut tube will be the peritoneal cavity. And these 3 cavities themselves coming from the intraembryonic coelomic cavity. But at the moment, let us discuss some of the derivatives coming from the ectoderm by drawing this diagram, a transverse section of embryo after gastrulation, talking about the ectodermal derivatives. Now, as we draw the transverse section of the embryo after gastrulation, we can see the 3 germ layers. The most dorsal is the ectoderm, and in the middle we'll have the mesoderm, which you can mark here. And then if we are looking towards the ventral side, it will be the endoderm. We also mentioned there will be a dorsal cavity, that is the amniotic cavity, and ventral is the yolk sac cavity, and a 3rd cavity develops, which will come on the lateral aspect in the mesodermal area. We are calling that cavity which is evident on the right side as well as left side as the intraembryonic coelomic cavity. So there are 3 cavities eventually. And we also mentioned the coelomic cavity itself will form 3 cavities, 2 in the thorax, 1 in the abdomen. Like in the thorax, the cavity will become pericardial cavity or pleural cavity, and in the abdomen it will become the peritoneal cavity. Now in the mesoderm, towards the center, there was an axial mesoderm. We were calling it as the notochord. And this notochord or the axial mesoderm is important because it is stimulating the overlying ectoderm, so that the overlying ectoderm changes into the neural plate ectoderm. Now, if there is neural plate ectoderm in the central portion, what is there on the periphery? Maybe on the right side, left side, there is surface ectoderm. Let us mark that as well. Here on the periphery is the surface ectoderm on either side. And what is this surface ectoderm supposed to contribute towards? There is a rule which we follow that any external opening in our body is lined by surface ectoderm. For example, the oral cavity at the beginning is lined by surface ectoderm. That is a rule. And in the oral cavity, there will be some glands forming. Like at the roof of oral cavity, we'll have the Rathke's pouch. And this Rathke's pouch is going to form anterior pituitary or adenohypophysis. In the oral cavity, we'll have the formation of salivary glands also coming from the same surface ectoderm. And salivary glands, as we understand, they are coming from the oral cavity, the parotid salivary gland, and the submandibular or sublingual salivary gland. These all structures are from the surface ectoderm. Surface ectoderm is also contributing to the skin epithelium and the glands like sweat gland, sebaceous gland. You can mention here surface ectoderm forming the skin epithelium, that is the skin epidermis, and also the glands like sweat gland, sebaceous gland. Now coming back to the other ectoderm which we were talking about, the developing nervous system. Neural plate ectoderm will form the neural tube and neural tube will form the central nervous system. You can mention it here. Neural plate ectoderm forming the neural tube, contributing to central nervous system which is the brain and the spinal cord. And then we talk about some cells here at the junction of the neural ectoderm and surface ectoderm to be called as the neural crest cells. Neural crest cells which are developing at the junction here were thought to come from the neuroectoderm earlier, but now it is mentioned the neural crest cells. They are actually the fourth germ layer coming from the epiblast cells directly, and these neural crest cells which are the fourth germ layer, they are contributing to the peripheral nervous system. Most of the ganglia come from the neural crest cells, and these neural crest cells along with the ganglia, as they contribute to peripheral nervous system, they are also forming secondary mesenchyme. As we see, the neural crest cells are contributing to the secondary mesenchyme, which is mostly restricted to the anterior lateral aspect of the head and neck region, basically in this region. And here it is contributing to most of the skull bones, the eyeball mesoderm, pharyngeal arch bones like the mandible bone, hyoid bone, and also the aorta pulmonary septum in the developing heart. So you can mention it is the neural crest cells which will form the secondary mesenchyme, will be contributing to the most of the skull bones, but especially on the anterior lateral aspect, because neural crest cells do not migrate posterior here. And then they are contributing most of the mesodermal component of the eyeball and the pharyngeal arch bones, like we have the mandible bone, the hyoid bone, and the aorta pulmonary septum in the developing heart. There's a condition called as DiGeorge syndrome, problem of the neural crest cell migration, resulting in skull defect, eyeball defect, pharyngeal arch defect, small mandible, and AP septum anomaly, aorta pulmonary septum anomaly, like tetralogy of Fallot, transposition of great vessels. And in this DiGeorge syndrome, it is observed that the problem of neural crest cell migration results in death of the baby, mostly because of AP septum anomalies. Cardiovascular defects will kill the baby. Now, these neural crest cells, they were contributing to secondary mesenchyme. What about the primary mesenchyme? The primary mesenchyme is here in the middle layer, and this mesoderm or the primary mesoderm is going to contribute to most of these structures in the body, because the secondary mesoderm is restricted to the anterior lateral aspect of the head neck region. If you're talking about occipital bone, neural crest cells do not form it. It is actually formed by the primary mesoderm. So you can mention it is the primary mesoderm which is going to form the occipital bone. And as we mentioned, since neural crest cells are restricted in this area, all the rest of the body, the mesoderm, is basically the primary mesoderm. It is the primary mesoderm which is contributing to skeletal muscles also. In this context, we can talk about the pharyngeal arch skeletal muscles like masseter. So masseter is a pharyngeal arch muscle formed by the primary mesoderm, not the secondary mesoderm. The neural crest cell forming secondary mesoderm will form the mandible bone, the pharyngeal arch bones, but not the muscles. Neural crest cells do not form the skeletal muscles. Skeletal muscles must come from primary mesoderm. So you can mention it is the primary mesoderm which is forming pharyngeal arch muscles like the masseter muscle. Whereas if you are talking about the mandible bone is a pharyngeal arch bone coming from the neural crest cell derived secondary mesenchyme. Now, neural crest cells are actually migratory cells and they'll contribute to a lot of ganglia. Let us enumerate some of the neural crest cell derivative in the next diagram. As we notice, it is going to form the cranial nerve ganglia, parasympathetic ganglia, or the sympathetic ganglia in the sympathetic chain. They also form the dorsal root ganglia. Plus, they are forming pre-aortic ganglia like celiac ganglion, mesenteric ganglion in front of the aorta. They also contribute to the airbags and mesonar plexus. Some of the parasympathetic ganglia, which are called enteric ganglia, maybe airbags ganglia or mesonar ganglia. They are all coming from neural crest cells. Most of the ganglia are from neural crest cell. The neural crest cell also contribute to sympathetic neurons in the adrenal medulla. And we are going to look at the neural crest cell derivative in the anterolateral aspect of the head neck region in the next diagram. As we mentioned, it is the neural crest cell forming the secondary mesenchyme. It will contribute to most of the eyeball component, which are mesodermal in origin. And along with that eyeball mesodermal component from the neural crest cell, it is contributing to the skull bones, basically the facial skeleton, though not the occipital bone. Occipital bone is mostly from primary mesenchyme, not from the neural crest cell. The neural crest cells also contribute to the aorta pulmonary septum, also called conotruncal septum. And if there is a problem, there can be pathology of Fallot or transposition gate vessels leading to death of the baby in DiGeorge syndrome. The neural crest cell will also contribute to pharyngeal arch bones like malleus incus stapes or mandible bone, the hyoid bone, but not the muscles. The pharyngeal arch muscles like masseter muscle come from primary mesoderm, not the secondary mesoderm. Then these neural crest cells in the head and neck region contribute to the skin dermis. So the skin dermis in this area is from mesoderm, the secondary mesoderm. But be careful. If you say epidermis, epidermis will come from the surface ectoderm. It is the dermis which will come from neural crest cell derived mesenchyme. It is the neural crest cell which will form the odontoblast and the dentine of the teeth. So odontoblast which will form the dentine of the teeth, or the teeth are from neural crest cell, but not the overlying enamel. If you are talking about the enamel, it is on the surface and it is from surface ectoderm. We said the oral cavity is lined by surface ectoderm. That surface ectoderm will form ameloblast. Ameloblast will form the enamel, and surface ectoderm also form the skin epidermis, as we have mentioned. So surface ectoderm form the enamel, the skin epidermis, and surface ectoderm will also contribute to the eyeball. The eye lens and first layer of cornea is from surface ectoderm. So in this discussion, we have talked about the surface ectoderm derivatives and the neural crest cell derivatives as well. Now we are going to talk about the derivatives of the mesoderm, and let us first draw a simple diagram before we move on to the detailed diagram for mesoderm. We already have mentioned that in a transverse section of the embryo there will be 3 germ layers. This time we are focusing upon the middle layer, the mesoderm derivatives, and there will be 3 cavities as well. The dorsal cavity is amniotic cavity, ventral is the yolk sac cavity, and then develops one lateral cavity which is intraembryonic coelomic cavity. So let us show that here in the region of the mesoderm. This is intraembryonic coelomic cavity. Then we mentioned that this intraembryonic coelomic cavity is further going to divide into 3 cavities, that is the pericardial cavity, pleural cavity, and the peritoneal cavity. So keeping that in mind, let us focus upon the axial mesoderm, which is in the center, the notochord, and then on the side, as we understand, para-axial mesoderm, and then we have got intermediate mesoderm, which will be in this region, and most laterally we will have what is called as lateral plate mesoderm. Let me zoom this area a little bit, and it is the lateral plate mesoderm where you have intraembryonic coelomic cavity. Because of that cavity, the lateral plate mesoderm itself divides into dorsal and ventral component. So now we are moving on to the detailed diagram here, and we can start with the notochord, the axial mesoderm. Now this notochord or the axial mesoderm, which we are showing here in the diagram we have drawn here simplistically, is the one which is contributing to nucleus pulposus of intervertebral disc. Then on the side you have para-axial mesoderm. That para-axial mesoderm is the one which is contributing to the somite, and this somite, as you see, is further going to divide into 3 components. We'll be talking about those 3 components called as the tomes. There is a sclerotome and a myotome and a dermatome. As the name is suggesting, this para-axial mesoderm, which is going to contribute to somite, will further form 3 components. Sclerotome means bone, like it will form the vertebral bone, and myotome, the skeletal muscles in the body, and dermatome, the dermis in the skin. And then after axial mesoderm, para-axial mesoderm, you have got intermediate mesoderm. That intermediate mesoderm is going to form kidney, testis, ovary, most of the genitourinary system. So here we mention this is the intermediate mesoderm mesoderm on either side, and it is contributing to kidney, testis, ovary. Most of the genitourinary system will develop from intermediate mesoderm on right side and left side. And then comes the lateral plate mesoderm. This lateral plate mesoderm on either side is being divided into a dorsal somatic and a ventral visceral, which is also called splanchnic mesoderm. Because of this cavity, we were talking about the cavity which is called intraembryonic coelomic cavity. So this is the intraembryonic coelomic cavity which we have mentioned is dividing the lateral plate mesoderm into dorsal somatic and ventral visceral lateral plate mesoderm. And as this cavity is contributing to pericardial cavity, pleural cavity or peritoneal cavity, their membranes, which is pericardium, pleura or peritoneum, will be derived from lateral plate mesoderm. So lateral plate mesoderm having two components. One is the somatic on the dorsal side, and there is the splanchnic or visceral lateral plate mesoderm on the ventral side. And as the name is suggesting, the ventral visceral will form the visceral layers of pleura, pericardium, peritoneum, whereas the dorsal somatic will be forming the parietal layers of pericardium, pleura or peritoneum. So the point is, as this cavity, which is coelomic cavity, itself is dividing into pericardial, pleural, peritoneal cavity, they have membranes. If it is the parietal peritoneum or parietal pleura or parietal pericardium, that will be coming from the dorsal somatic lateral plate mesoderm, whereas the ventral, visceral or splanchnic layer lateral plate mesoderm is contributing to the visceral pleura or pericardium or the peritoneum. But then they have some other contributions as well. Like we have to mention the dorsal somatic lateral plate mesoderm is going to contribute to appendicular skeleton, upper limb, lower limb bones, and the ventral visceral lateral plate mesoderm is contributing to visceral muscles, the splanchnic muscles, that is cardiac muscle and the smooth muscles. So cardiovascular tubes develop from here. Ventral visceral lateral plate mesoderm forms the cardiovascular tube. The cardiac muscles, smooth muscles develop here for most part of the body. Now we said that the upper limb and lower limb bones, the appendicular skeleton, is developing from the dorsal somatic lateral plate mesoderm. What about the vertebra? The vertebra develop from the sclerotome in the paraaxial mesoderm. Then what about the skull bones? And we have mentioned if it is the anterior lateral skull bones, they will come from neural crest cell derived secondary mesenchyme. So let us look at a diagram to talk about the skeleton of the body, which should come from the mesoderm. And as we observed, anterolateral part of the skull, especially the facial skeleton, is coming from neural crest cell derived secondary mesenchyme. But then we know that the posterior bone, the neural crest cells are not here. The posterior part of the occipital bone and the vertebra, entire vertebra, they are all coming from paraaxial mesoderm. And that paraaxial mesoderm, which forms the vertebra, is basically primary mesenchyme. These vertebra, they are basically coming from the somites, which is developing in the paraaxial mesoderm, and it is primary mesoderm. Then what about the upper limb and lower limb bones? If we are talking about the upper limb bones or the lower limb bones, that is the appendicular skeleton. And appendicular skeleton come from lateral plate mesoderm. If you say lateral plate mesoderm, which portion? The dorsal somatic or ventral visceral? See, these bones, they are in the periphery. They're not visceral, they are somatic. So dorsal somatic lateral plate mesoderm. It is the dorsal and somatic lateral plate mesoderm which will form the appendicular skeleton, which is the upper limb and lower limb bones. All the discussion is based upon the type of questions we get. Now we are supposed to move on the details of the somite, the paraaxial mesoderm. We have mentioned the somite itself will have three components: the sclerotome, myotome and dermatome. It was the sclerotome which was forming the vertebra. Then what about the other two components? Let us look at the diagram. And once again, we can see this is the axial mesoderm, the notochord, and then this is the paraaxial mesoderm, the somite. And this somite itself is giving three components, the three tomes. The middlemost is the sclerotome, which will form the vertebra. The middle one is the myotome, which will form the skeletal muscle of body, and dermatome, which is going to form the skin dermis. As you can see, the most lateral, the dermatome forming the dermis of skin, and the middle, the myotome, is forming the skeletal muscle of the body, and the most medial sclerotome will form half vertebra here and half vertebra here, fuse to become one vertebra and then cover the neural tube. In this case, it is the spinal cord, which is developing from the neural tube, which itself is coming from neuroectoderm. So, as you can mention, we are talking about the somite, which is paraaxial mesoderm. And this paraaxial mesoderm or the somite has three components. It is the middlemost, that is the sclerotome, which is contributing to half of the vertebra, and then they fuse to become one vertebra. So vertebra is derived from the sclerotome, and the middle is the myotome, which will form the skeletal muscles. It's a statement by Gray's Anatomy. All the skeletal muscle in our body will be developing from the myotome part of somite, and then the lateralmost, which is the dermatome, is going to form the dermis of the skin of the somite or paraaxial mesoderm, which is primary mesoderm. Secondary mesoderm coming from neural crest cell in the anterolateral aspect of the head and neck region, contributing to facial skeleton chiefly, and pharyngeal arch bones like mandible bone, head bone. They are all from neural crest cells, but not the posterior part of occipital bone. Posterior part of occipital bone must come from the paraaxial mesoderm or the primary mesoderm. In this diagram, we are going to talk about the endodermal derivatives, and the very first thing you have to talk about is the gut tube. But along with the gut tube, we also find that respiratory tube is developing from the endoderm. And even the lower part of genitourinary tube is also coming from the endoderm. So three tubes basically: the gut tube, respiratory tube, and lower part of genitourinary tube. As we see, the gut tube is divided into three components. There is a foregut here, then we have got the midgut here, and then the hindgut component of the gut tube. In the terminal hindgut, we have the cloaca region, where develops one urorectal septum, dividing the posteriorly placed rectum from the anteriorly placed urogenital sinus. So here we will have the urorectal septum, and this urorectal septum, as the name is suggesting, is present in the cloaca region of the hindgut, separating rectum and anal canal posterior and urogenital sinus anterior. It is the urogenital sinus, as the name is suggesting, will contribute to urinary bladder, urethra, and lower portion of the vagina. So it is the urinary bladder and urethra and lower vagina, which are also endodermal in origin. Basically, their epithelium coming from urogenital sinus. And then we see one diverticulum, which is called as the allantois. It is a hindgut diverticulum, which you see here, coming from the urogenital sinus, apical portion, the apical portion of urinary bladder. This allantois is endodermal diverticulum from the hindgut, becoming a content of the umbilical cord. Now, this is hindgut diverticulum. There is a midgut diverticulum also, becoming a content of umbilical cord. We call it as vitalointestinal duct. As you can show, this is the midgut diverticulum, vitalointestinal duct. Normally, the allantois as well as the vitelline duct must get obliterated in the normal course of development. Now, in the hindgut region, we have urorectal septum. In the foregut region, we have tracheoesophageal septum, and that tracheoesophageal septum is separating trachea anterior and esophagus posterior. And from the tracheal diverticulum, we have trachea bronchus and lungs, the respiratory tube developing from the gut tube, that is foregut tube. And we understand anteriorly is the trachea bronchus lung, posteriorly is the esophagus. Between the two, we'll have tracheoesophageal septum in the foregut region. Now, in the lateral wall of the pharynx, that is foregut derivative, we have some pharyngeal pouches, and these pharyngeal pouches, which we see developing in the lateral wall of the pharynx, will be contributing to some of the glands, like the thymus gland and the parathyroid gland. These thymus gland, parathyroid gland, they are parenchyma, the epithelial component. The glandular tissue is coming from the endoderm of the pharyngeal pouches. Now, there are two more glands, that is the liver and pancreas, developing at the junction of foregut and midgut, and they are endodermal origin, mostly derived from the foregut and partly from the midgut. They are lying at the junction of the foregut and the midgut. So here we have discussed the endodermal derivatives. Basically, the epithelial lining and the glands coming from that epithelium, they are all from the endoderm. Now let us talk about the extra embryonic structures as well. We have been discussing about the embryonic ectoderm, mesoderm, endoderm, but now we are going to talk about the extra embryonic structures, which are going to contribute to structures like the placenta and the placental membrane, the chorion, the amnion. And as you can see, The placenta is contributed by the trophoblast. Tropho means nutrition. Placenta is for the nutritional purpose. And then you can see something written as extra embryonic mesoderm. So outside the embryo. Now a question is coming. It is asking, where is this extra embryonic mesoderm derived from? And it is a controversy. The answer we should give is the cells which line the primary yolk sac or primary umbilical vesicle, the other name, will be contributing to extra embryonic mesoderm, which will be contributing to placental membranes like the amnion and chorion. So primary yolk sac or primary umbilical vesicle is lined by some cells, and those cells are contributing to extra embryonic mesoderm. Though there is a controversy, as I mentioned, some books will say extra embryonic mesoderm, which you are seeing here, is derived from the hypoblast. Some books will say extra embryonic mesoderm is derived from the epiblast. And there's also description of this extra embryonic mesoderm coming from the trophoblast. So answer could be trophoblast, epiblast, hypoblast, but the best answer is cells lining the primary yolk sac or primary umbilical vesicle. That is the one going to form extra embryonic mesoderm. And then we know that trophoblast, along with this extra embryonic mesoderm, will contribute to placental membrane, the chorion and the amnion. Before we start discussing that, if you remember, we have discussed in the embryo, in the beginning there are 2 cavities, then there are 3 cavities. There is a dorsal amniotic cavity, ventral yolk sac cavity, and then a lateral cavity, coelomic cavity. But that was intra-embryonic coelomic cavity. There's a description of extra embryonic coelomic cavity as well, because this time we are talking about extra embryonic structures. So this mesoderm, which is extra embryonic mesoderm, will develop a cavity, and that cavity is called extra embryonic coelomic cavity, dividing the extra embryonic mesoderm into 2 components. The one which is towards the yolk sac is called as the visceral, and the one which is towards the outside periphery, parietal, that will be somatic extra embryonic mesoderm. This will become evident in the next portion of the diagram. As you see, we are talking about the cavity called as extra embryonic coelomic cavity. And this extra embryonic coelomic cavity develops in the extra embryonic mesoderm, dividing into 2 parts. The extra embryonic mesoderm, which is along with the yolk sac, is called as the splanchnic or visceral layer of extra embryonic mesoderm, whereas the one which is at the parietal or periphery, it is called somatopleuric extra embryonic mesoderm. So as you magnify, you find extra embryonic mesoderm dividing into 2 components. And as we focus here, the mesoderm which is towards the yolk sac is of course inner or visceral or splanchnic. So this is the splanchnic extra embryonic mesoderm, whereas rest all, which you can start from here and going around, is the parietal or peripheral or somatic extra embryonic mesoderm. Let us mark that as well. This is the parietal, peripheral or somatopleuric extra embryonic mesoderm. And as you follow it all around the developing embryo, you will find that it is coming from periphery towards the interior as well, and then covering the amniotic cavity. So covering the amniotic cavity and the extra embryonic coelomic cavity is the somatopleuric layer of extra embryonic mesoderm. That means only the yolk sac is covered by a special extra embryonic mesoderm, which is called splanchnopluric. Only that much of the mesoderm lining the yolk sac, which is now called secondary yolk sac. So yolk sac is primary, secondary and tertiary. We'll have a diagram to discuss the 3 types subsequently, but at the moment you'll find that the lining of amniotic cavity is called as amnion, and amnion is having this parietal extra embryonic mesoderm plus some cells lining the roof of amniotic cavity called amniogenic cells. So amniogenic cells plus extra embryonic mesoderm, which is somatopleuric extra embryonic mesoderm, the amnion, and this amnion is going to line the amniotic cavity. Then what about the chorion? The chorion is on the outside and will have this extra embryonic somatopleuric layer, but along with that it is also having the trophoblast. So chorion is the placental membrane towards the exterior, and you can see it is contributed by the trophoblast and again the extra embryonic mesoderm, which is somatopleuric. And we can also see there is a connecting stalk, which will later become primary umbilical cord. So what we have learned here is it is the extra embryonic coelomic cavity which is outside the embryo, which has divided the extra embryonic mesoderm into 2 components. The one towards the yolk sac is the splanchnopluric and does not contribute to any of the chorion or amnion membrane. It is the parietal or somatopleuric layer which contributes to not only chorion but also the amnion. Talking about the placenta will have two components. One is maternal and one is the fetal. So maternal placenta is contributed by the decidua bacillus, that is the functional endometrium of the uterus. And when it comes to the fetal placenta is contributed by the chorion, sending some finger-like processes, the chorionic villi, and forming what is called as chorion frondosum. So this chorionic villi or chorion frondosum is the fetal placenta. Now, this is the fetal placenta and decidua bacillus is the maternal placenta. Both of them will be supporting the metabolic requirement of the embryo, maybe the respiratory gaseous exchange or excretion of nitrogenous waste products. That is the function of the placenta. Then we have to talk about the primary, secondary and tertiary yolk sac as well. In this diagram, we can discuss the primary yolk sac becoming the secondary yolk sac, becoming the tertiary yolk sac. It is understood that in the beginning there is a dorsal cavity, amniotic cavity, and ventral is the yolk sac cavity. That time we call it as the primary yolk sac. And then we find that there is a cavity developing outside the embryo. We call it as the extra embryonic coelomic cavity. And once the extra embryonic coelomic cavity is developing, the primary yolk sac becomes secondary yolk sac. So here it is the secondary yolk sac, which will then become the tertiary yolk sac. The tertiary yolk sac actually becomes very small and is continuous with the gut tube. The gut tube is lined by endoderm and so is the yolk sac lined by endoderm. The yolk sac becomes very small and is continuous with the midgut with the help of a midgut diverticulum called as vitalo-intestinal duct, which is becoming a component of the umbilical cord. In the umbilical cord, we also have a hindgut diverticulum which is called as allantois. So let us zoom this diagram, and as you can notice one more thing, because there is cephalocaudal folding of embryo happening. That means the head end of the baby and the tail end of the baby, they are coming together. Cephalocaudal folding. In that process, the amniotic cavity surrounds the embryo all around. And because the amniotic cavity is becoming enlarged, the yolk sac cavity is becoming small. Tertiary yolk sac becomes very small and ultimately it will regress. This connection with the midgut vitalo-intestinal duct will also regress. Even the hindgut diverticulum allantois will regress inside the umbilical cord. And this is the placental area where we have mentioned the fetal placenta is chorion frondosum and the maternal placenta is the decidua bacillus. This area will be lined by chorion, where you have trophoblast plus somatopleuric extraembryonic mesoderm. Because this somatopleuric extraembryonic mesoderm is also contributing to the amnion, which is covering the amniotic cavity. This amnion not only covers the amniotic cavity, it also lines the umbilical cord and become continuous with the chorion membrane. The chorion, which contributes to fetal placenta, chorion frondosum. These diagrams you can see once again in the next coming slides. As we revise the details again, we can see this is the conceptus which is implanted into the uterine endometrium, the functional endometrium, which is called as the decidua bacillus, contributing to the maternal placenta. But then what about the fetal placenta and the placental membrane? See, in the beginning, we have a dorsal amniotic cavity, ventral is the yolk sac cavity, and this is the primary yolk sac. And it was the lining of the primary yolk sac which has contributed to extra embryonic mesoderm. So you can see this is the primary yolk sac cavity, and the cells which were lining it will give the extra embryonic mesoderm, which is two types. If it is lining the yolk sac, you will call it as visceral or splanchnic, which we can shade here, and the one which is not contributing to any of the amnion or chorion. But if you are going beyond that, may it be lining the amniotic cavity or the connecting stalk, which is the umbilical cord, or in the periphery, this extra embryonic mesoderm in the periphery. This is all peripheral or parietal or somatic extra embryonic mesoderm, and it is the one which will contribute to chorion and amnion. So what is amnion then? Amnion is basically lining the amniotic cavity, some amniogenic cells, and the extra embryonic mesoderm of the parietal variety. So this is the amnion, the placental membrane. What about the chorion then? If we are talking about the chorion, then this is the chorion, and in the chorion we have to include not only the extra embryonic mesoderm, which is the parietal or somatic layer, but along with that, the trophoblast as well. Towards the baby, it is cytotrophoblast, and towards the maternal side, it is syncytial trophoblast. So, as we can see, it is the exterior syncytial trophoblast and the interior cytotrophoblast, together with the extraembryonic mesoderm, will be contributing to the chorion. Chorion is the placental membrane, which will be then forming the primary villus, secondary villus, tertiary villus, the chorion fundosum, the fetal placenta. Now we can move on to the next diagram in sequence. Here, once again, we can see the extraembryonic membranes, that is the amnion and the chorion. Amnion is lining the amniotic cavity, which is now becoming larger, trying to surround the embryo from all around, and it is contributed by amniogenic cells plus extraembryonic mesoderm. You can mention here the amnion is amniogenic cells plus extraembryonic mesoderm, which is parietal layer. And what about the chorion? Chorion is extraembryonic mesoderm of the parietal category, plus it is the trophoblast. Trophoblast is two types, the cytotrophoblast and syncytiotrophoblast. So as you can see, this is the chorion, which is having the extraembryonic mesoderm, the parietal category, plus the cytotrophoblast and syncytiotrophoblast, forming the primary, secondary, tertiary villus and chorion fundosum, the fetal placenta. One other thing what we notice is the head end of the baby folds towards the tail end of the baby, cephalocaudal folding. Because of that, the amniotic cavity is enlarging and surrounding the baby, whereas the yolk sac cavity is getting incorporated into the baby, contributing to foregut, midgut, and the hindgut. And eventually, the yolk sac will become very narrow, and then it regresses, which can be seen in the next part of the diagram. In this diagram, we find that as the baby is folding head to toe, the cephalocaudal folding, the amniotic cavity becomes larger and larger, whereas the yolk sac cavity becomes very small. This is going to be the tertiary yolk sac, and ultimately it regresses. It is in continuity with the midgut, which is called as the vitellointestinal duct, the midgut diverticulum, which is going to become a component of the umbilical cord here. In the umbilical cord, we also have the hindgut diverticulum, which is the allantois. But later, the allantois or vitellointestinal duct or the yolk sac, they all digress, and what is left is just the foregut, midgut, and the hindgut. And in this diagram, you can see the uterus and the baby in entirety. The amniotic cavity has almost surrounded the embryo from all sides, and the amniotic cavity is itself covered by the placental membrane, the amnion. You can see here the amnion is lining the amniotic cavity all around, covering the baby, suspended in the amniotic fluid. And the amnion is also lining the umbilical cord, which was initially called connecting stalk, and this time it is primitive umbilical cord. And then the amnion becomes continuous with the chorion. The chorion, which is contributing to chorion fundosum or fetal placenta. So let us trace the chorion as well. And as you do that, we'll find that amnion is becoming continuous with the chorion which lines the fetal placenta, that is called chorion fundosum, and is surrounding the baby as well. So this is the chorion. And the chorion was having the finger-like processes. That is the primary, secondary, tertiary villus, contributing to fetal placenta, which is called as chorion fundosum. So here it is the fetal placenta, the chorion fundosum, which is in contact with the maternal placenta, that is the decidua vessalis, and taking care of the metabolic requirement of the baby. May it be the respiratory gaseous exchange or the excretion of the metabolic products. Now let us talk about the primary, secondary, tertiary villus in detail. And as we notice, the description is for primary, secondary, and the tertiary villus. In the primary villus, which is formed around day number 12, you'll have the inner cytotrophoblast and the outer syncytiotrophoblast. Towards the maternal side is syncytiotrophoblast helping in implantation. Then comes the extraembryonic mesoderm, and the villus becomes secondary. And as we reach the third week, there is a tertiary villus coming, where you have fetal blood vessels also evident. We have got one more diagram to discuss the same, and as we see, if it is the primary villus, it is day number 12, having inner cytotrophoblast and outer syncytiotrophoblast towards the maternal side. And when there is extraembryonic mesoderm coming, we call it as the secondary villus. And if we have the fetal blood vessels coming in, then it becomes tertiary villus in the week number 3, day number 17 to 21. Actually, that is the time when there will be fetal placental circulation beginning. So fetal placental circulation begins on day number 17 to 21, where the villus is having inner core of extraembryonic mesoderm with suspended fetal blood vessels. There is one fetal placental circulation and there is one uterine placental circulation. Remember, the uterine placental circulation begins at day number 12, where the uterine artery is supplying the blood towards the maternal placenta, the uterine placenta. That is why it is called uterine placental circulation. But once the blood reaches the fetal placenta also, then you start calling it as fetal placental circulation, which is a bit later. That is in the week number 3, when you already have formation of the tertiary villus. We are beginning with the histology section now, and in the first discussion, we'll be looking at the type of epithelia. The epithelium can be simple or stratified. Simple means it will be single cell layer and stratified means it will be multiple cell layer. And when we talk about the simple or single cell epithelium, there are 3 types to be discussed. As we see in this diagram, it could be a flat cell with flat nucleus and it is called as the simple squamous epithelium. As you observe, it is a flat cell with flat nucleus. But then not necessarily the nucleus can be actually globular sometime, as in this cell it is appearing globular. So you can mention could be a flat nucleus or globular nucleus. Then coming to simple cuboidal epithelium, the name is suggesting it is cuboid shape. That means the height of the cell is equal to breadth of the cell. And you will notice that along with the fact that height is equal to breadth, there is a central spherical nucleus. Comparatively, in the simple columnar epithelium, we find the cells are column shaped, means the height is more than the breadth and there will be oval basal nucleus. So we can mention the details here. Simple columnar epithelium, the height is more than the breadth of the cell and there are oval basal nuclei. Plus one more thing you can notice. In case of simple squamous epithelium, there is only a thin rim of cytoplasm above the nucleus. Whereas in the cuboidal epithelium, there is good amount of cytoplasm above the nucleus because the nucleus is central. And then comparatively in the simple columnar, the amount of cytoplasm of the nucleus is like a column. So you can compare the columnar cell epithelium with the cuboidal and the simple squamous with the amount of cytoplasm above the nucleus. As you can notice, here it is a thin rim of cytoplasm. But in cuboidal, the cytoplasm is increased above the nucleus. Whereas in the columnar, you can see there is abundant cytoplasm above the nucleus. Now let us take some examples, the sites where these type of epithelia are present. And starting with the simple squamous epithelium, we observe them in the mesothelia. Mesothelia is the membrane which will be lining the 3 cavities, pericardial, pleural and peritoneal cavity. So lining membrane, the peritoneum or the pericardium or the pleura, they have simple squamous epithelium which is called as the mesothelium. And you'll notice that it is present in the endothelium also. That could be the heart or that could be the blood vessels and even the endothelium of the 5th layer of cornea, the inner epithelium, the endothelium. And this type of epithelium can also be seen in the kidney tubules like the loop of Henle. And we understand type 1 pneumocyte of the alveolus is also the simple squamous epithelium. Now here in the diagram we are looking at the peritoneum, which is a type of epithelium belonging to mesothelia and flat cell with flat nucleus. As you can observe, there is very thin rim of cytoplasm above the nucleus. Normally the nucleus should be flat nucleus, but as I mentioned, it could be globular also at certain occasions. Now let us move on to the examples of simple cuboidal epithelium as well. And there, for simple cuboidal epithelium, the glands have this kind of epithelium, even the ducts, though the ducts are usually stratified cuboidal epithelium. Then in the renal tubule, like PCT, DCT, you will have simple cuboidal epithelium. And along with that, if we are looking at the thyroid follicles also, they have simple cuboidal epithelium. In this present diagram, we are looking at the kidney, the uriniferous tubule, the nephron, and the DCT, distal convoluted tubule. As you will notice, it is having simple cuboidal epithelium. The cells are having central spherical nucleus, and height of the cell is equal to breadth of the cell, like a cuboid. That is why they are called as cuboidal epithelium. And let us now move on to the examples of columnar epithelium as well, which you will find at various sites like the GIT, starting with the stomach, then in the intestine, maybe small or large intestine, and then even the gallbladder. You'll find that in the gallbladder there are some microvilli. Even in the small intestine, we will have the microvilli. And in the small intestine, the microvilli can give you the striated appearance. So your eyes must be trained to identify. In the exam, if there's a slide given and you have to identify the microvilli, they will appear like some eosinophilic apical modification, the striated border in the small intestine. Tall columnar cells with oval basal nucleus. Now you can observe the nuclei may not be basal all the time, but one thing is sure. Maybe the nuclei are not appearing basal all the time, but there is abundance of cytoplasm above the nucleus. So as you can see, it cannot be squamous, it cannot be cuboidal. It goes more in favor of the columnar epithelium. Again, you have to train your eyes for comparative analysis and identification. And this kind of epithelium can also be found in the collecting ducts of the kidney, where few of the cells can have microvilli again. But those microvilli in the collecting ducts are very short and very few in number. All of these are MCQs. Then we look at the stratified epithelium, where there are multiple levels of cells showing multiple levels of nuclei. Hence, we are calling it as multilayered or stratified. Now, the very first diagram is stratified squamous epithelium. As it is understood, the basal cells are columnar. In the middle, we have the cuboidal cells. Only the superficial cells are having flat nucleus, and they are the squamous cells. But the squamous cells at the superficial layer gives the name stratified squamous epithelium, and you can see some keratin also on the apical surface. So this kind of appearance will be present in the skin, the stratified squamous keratinized epithelium. Then coming to the next diagram, you can see again there are multiple levels of nuclei, but the topmost cells towards the surface apical region, the cells are not flat, they are large globular cells, and that is how appears the transitional epithelium, the urothelium, the lining of the urinary tube like urinary bladder or the ureter. So how do you differentiate the urothelium from the stratified squamous epithelium? One of the fact is that the terminal cells towards the apex in keratinized epithelium were very flat cells with flat nucleus, and that kind of flat nucleus appearance is not there in the urothelium, because in the urothelium there are large globular cells with large globular nucleus, and these cells are basically so large that, as you see, they give the appearance of umbrella shaped cell. Like this is an umbrella, one single cell, and under that numerous cells are hiding. As if there is rain going on, an umbrella is taking care of these many cells under its cover. So umbrella shaped cell, that U represents the umbrella shaped cells. They are stretchable, and that stretchability of the umbrella cells help in storage of urine. And when it comes to the pseudostratified ciliated columnar epithelium, first of all, you can notice the cilia. Your eyes should be able to identify the appearance of cilia. So these are fine thread-like processes. The C is representing the cilia. Plus, this is an epithelium with multiple nuclei at multiple levels, upper, middle, lower. So giving the wrong impression of being stratified, but it is not stratified, since each of the cell is touching the basement membrane. So it cannot qualify as stratified, but appearing like stratified. That is why the name comes pseudo-stratified, giving false impression of being stratified. And they are columnar cells, as you will find in the respiratory epithelium. Like the laryngeal part of epiglottis comes as a question, it will have respiratory epithelium. The pseudostratified ciliated columnar epithelium. We are looking at few more sites for discussing the type of epithelium, and for that we have taken one sagittal section of the head and neck region, looking at the two tubes which develop together, the respiratory tube and the gut tube. Now remember one simple rule for any external opening in our body. Any external opening in our body is lined by stratified squamous epithelium. Say for example the oral opening or the nasal opening, the first layer of cornea, the external ear ear canal, all these openings at the outer surface, they are lined by stratified squamous epithelium. Say, for example, here, the nasal vestibule, where the air will enter. So as you are marking the nasal vestibule, this area is lined by true skin with hair, that is stratified squamous epithelium, which is keratinized. The same type of epithelium not only is found at the nasal opening, nasal vestibule, but also the oral opening, even the hard palate and the dorsum of the tongue in the initial part of oral cavity. As you mark them, they are lined by the stratified squamous epithelium, and which is keratinized. But then when you are moving from hard palate to soft palate, the epithelium will change. It will become non-keratinized stratified squamous epithelium. So we miss the keratin here in the deeper part. Even the posterior part of tongue will be non-keratinized. So which all areas are lined by the non-keratinized stratified squamous epithelium? They are the soft palate, the posterior part of the tongue, and the tonsils also, maybe palatine tonsil or lingual tonsil. And then when you are looking at this lid over the larynx, which is epiglottis, the lingual part of epiglottis. See, this is the epiglottis, which is lid over the larynx. It will have two surfaces. One is laryngeal surface and one is oral surface, the lingual surface. So lingual surface of epiglottis is lined by non-keratinized stratified squamous epithelium, same as the soft palate or the posterior part of the dorsum of the tongue or palatine tonsil, lingual tonsil. So what is the difference between this epithelium and this epithelium? Actually here, which is the hard palate and dorsum of the tongue in the initial part, keratin is there. Keratinized epithelium is there. Now this non-keratinized stratified squamous epithelium can also be seen in the true vocal cord, which you can mark here. The true vocal cords in the larynx will also be lined by non-keratinized stratified squamous epithelium. Otherwise, when we talk about the respiratory tube, may it be the larynx or the laryngeal part of epiglottis or the adenoids, that is the pharyngeal tonsil. The respiratory tube, most of it, say the nasal cavity, paranasal sinuses, the nasopharynx, the adenoids, which we can mark here, they are all lined by respiratory epithelium. Pseudostratified ciliated columnar epithelium is the respiratory epithelium, which is found in the nasal cavity, in the nasopharynx, the adenoids, or the laryngeal part of epiglottis, as we are putting it here, and even throughout the larynx, it is respiratory epithelium, but except the vocal cord, the true vocal cord, because there you have non-keratinized stratified squamous epithelium. We have a diagram to discuss the details of larynx and epiglottis in the next slides. And here we have magnified the diagram of epiglottis, the lid over the larynx to close the larynx. It has two surfaces. One is towards the tongue, the lingual surface lined by non-keratinized stratified squamous epithelium, and the other surface, which is towards the larynx, the laryngeal surface lined by respiratory epithelium, the pseudostratified ciliated columnar epithelium. So, as you can see, we are magnifying the lingual surface towards the tongue, and as it is observed here, it is non-keratinized stratified squamous epithelium. So we are looking at the lingual surface here having non-keratinized stratified squamous epithelium. But if we are looking at the laryngeal surface, as we will demonstrate now, this laryngeal surface towards the larynx of the epiglottis is lined by the respiratory epithelium. As is evident, the epithelium toward the larynx is the pseudostratified ciliated columnar epithelium with goblet cells, and we have to remember epiglottis is having a core of elastic cartilage with a lot of elastic fibers and chondrocytes. Though respiratory tube is made up of hyaline cartilage, but there are some exceptions, like epiglottis does not have hyaline cartilage. It is having elastic cartilage. But if you're looking at the larynx, like here we have magnified the view of larynx, and the laryngeal cartilage is mostly hyaline cartilage. You can see here a lot of chondrocytes in glass-like the hyalos matrix, so hyaline cartilage. And along with the hyaline cartilage of the larynx, you can see the vocal cord area also. We understand the larynx is all lined by respiratory epithelium, but except the true vocal cord, as they are prone to abrasion due to fast-moving air, epithelium resistant to abrasion is stratified squamous epithelium. So as you look at the true vocal cords, only this area is lined by stratified squamous epithelium. Otherwise, rest of the larynx, as you will see, is having respiratory epithelium, which is pseudostratified ciliated columnar epithelium with goblet cell. So only this area of the true vocal cord, which you can mark now here, is lined by the non-keratinized stratified squamous epithelium. But then we have to remember this kind of epithelium, which is in the vocal cords, can encroach upon the other areas also in a chronic smoker, squamous metaplasia. And if there is squamous metaplasia, replacing the respiratory epithelium with the squamous epithelium, metaplasia can become dysplasia, can become cancer. So CA larynx, which is obviously a dangerous situation. Continuing with the type of epithelia, and this time we are looking at the various body tubes. If we are starting with the esophagus, we'll find that it is having non-keratinized stratified squamous epithelium. But as we move towards the stomach or the intestine, maybe small intestine or large intestine, the stomach and intestine will have simple columnar epithelium. But specifically, if we are talking about the small intestine, it will have three modifications. That is to increase the surface area of absorption, the plicae circulares, mucosal folds, and the villi and microvilli. These three modifications are present only in small intestine, will be absent from large intestine and also the stomach. Now, as we look at the respiratory tube, like the trachea and the bronchus, will have the respiratory epithelium, pseudostratified ciliated columnar epithelium with goblet cells. And as it comes to the renal tube, we already know, may it be the urinary bladder or the ureter or the proximal urethra, will have the transitional epithelium, the stretchable epithelium, as we mentioned, umbrella-shaped cells towards the luminal side. Now, coming to the skin epithelium, will be stratified squamous epithelium, but with keratin. If there is a dry surface, you'll have keratin. And though this epithelium, which is stratified squamous epithelium, is very similar to the esophagus, but esophagus is a wet surface, so it will have non-keratinized stratified squamous epithelium. Skin is keratinized, keeping the surface dry. In fact, all these body tubes, they are wet surfaces and they should be without keratin. Now, when we are talking about the body tubes, they'll have usually 4 layers. Lining the lumen, there'll be mucosa. Under that, there is submucosa, then muscular externa, and the outermost, the connective tissue layer, which is called adventitia. But if you have a lining of the peritoneum, like in stomach or intestine, we will call it as serosa. So peritoneal serosa will have simple squamous epithelium, which is also called mesothelium. And then there is some arrangement of smooth muscles, the muscular externa, in the gut tube. In the gut tube, that is esophagus, stomach or intestine, there is inner circular layer and outer longitudinal layer of smooth muscle. But that is exactly reverse in case of renal or genital tube, because in the renal and genital tube, the inner layer is longitudinal and outer layer becomes circular. But be careful, these body tubes can have an additional third layer at occasions. Then in the respiratory tube, we have additional hyaline cartilage to maintain the rigidity and maintain the patency of the lumen, maybe the trachea or the bronchi level. And between the cartilage and the respiratory epithelium, there are some smooth muscles. These smooth muscles can help us in narrowing the lumen of the respiratory tube, which is called bronchoconstriction under the autonomic nervous system. Bronchodilation, bronchoconstriction, as is carried out by the smooth muscle cells. We understand that the epithelium can modify itself as gland and it can give some secretion. According to the type of secretions formed by the epithelial cells, we can classify the glands into 3 types: the merocrine, apocrine, and the holocrine variety. As we understand in these diagrams, if we are looking at the salivary glands like parotid salivary gland, it is merocrine or acrine variety of glandular secretion. When it comes to the mammary gland, it is modified sweat gland of apocrine variety. Apocrine, the apical surface of the cell is broken and there is loss of apical cell membrane, which is then going to cover the secretion, which is going to form some bleb. So that is the apocrine variety, whereas in the holocrine variety, whole of the cell membrane is broken. In fact, whole of the cell is broken and there is total loss of cell membrane. The organelles are also broken down to form the secretion, like the sebum formed by the sebaceous gland associated associated with hair follicle in the skin. Now we have got some histological pictures to describe the merocrine, the apocrine, and the holocrine glands once again. So here we can see this is the merocrine, or what is also called acrine variety of gland, whereas this is the apocrine variety, and here is the holocrine. Let us zoom this diagram of what is called as the merocrine or acrine. Now in this merocrine or the acrine variety of the glands, the secretions are coming into the lumen, but there are intact cell membrane. The cell membrane is not lost. As you compare it with the apocrine variety, you will find there is loss of apical cell membrane, and which is then covering the secretions given by the cells, forming some blebs. So these are the cells which lose their apical cell membrane, which is covering the secretions to form the bleb. So loss of apical cell membrane, the apocrine variety of glandular secretion. Like we saw, it is the mammary gland, modified sweat gland of apocrine variety. And when we are looking at the sebaceous gland, it is holocrine variety. Whole of the cell membrane, even the cell organelles, are lost. This is the hair follicle in the skin, and associated with hair follicle, you can see these are the sebaceous glands. And if we look at the peripheral portion of the gland, there is intact cell membrane. But as we move towards the duct portion, there is loss of cell membrane and the cell organelles, entire cellular content becoming the sebum, which is poured along the duct alongside of the hair follicle. So sebaceous gland is one holocrine variety of the glandular secretion. We have one diagram of the skin as well, showing again the sebaceous gland details. And as we notice, this is the skin epithelium. Along with that, we can also see this is the hair follicle. And along with the hair follicle, we find there will be a duct of sebaceous gland. So this is the sebaceous gland where the peripheral cells are having intact cell membrane, but towards the secretory portion, loss of cell membrane, cell organelles, all becoming sebum poured alongside of the hair follicle to keep it lubricated. So this is holocrine variety, the sebaceous gland found along with hair follicle. But if you look at this region here, this is the sweat gland, mostly the merocrine or acrine variety of secretion, and the cellular lining, a simple cuboidal epithelium. The ducts can become stratified cuboidal epithelium. So sweat gland is mostly the merocrine variety, whereas the sebaceous gland is holocrine variety of gland. This is another type of classification for the type of glands. As we can see, there could be serous type of glands, or there can be mucinous type of gland, or there can be a combination which is called seromucinous type of gland. Now, as we are talking about the salivary glands around the ear, the parotid salivary gland is predominantly serous type of gland. But if we are looking at the salivary gland which is under the tongue, the sublingual salivary gland, then it is predominantly the mucoid type of gland. So sublingual is mucus type of gland. Then when it comes to the gland under the mandible, that is submandibular salivary gland, it is seromucinous or mixed salivary gland. It is called mixed because it is having some serous acini and also the mucinous acini. Actually, it will have what is called as the serous demilume as well. Now, before we zoom this area, it has to be understood that parotid salivary gland or the serous type of glands, they will have thin, watery secretions as compared with the sublingual salivary gland, which is mucoid type of gland. It will have thick and viscous type of secretions. And as we zoom here, you can see the parotid salivary gland as compared with the sublingual is having the serous cells which are dark staining, means they will take more of the eosin, so they will become dark pinkish as compared with the mucinous type of cells which are lightly staining, and due to presence of the mucoid secretion, cells have empty look, foamy appearance. So this is about the sublingual salivary gland. The mucous cells are light staining and they are having some empty look or foamy appearance because of the mucoid type of secretion. Now, a combination of the two which we are talking about, submandibular salivary gland, you can see this is seromucinous gland. There are serous cells, there are mucous cells, and then we can see what is called serous demilume. There is a cap of the serous gland which is sitting on the mucous gland. gland. Like here you see, this is the mucous gland, lightly staining cells, and darkly staining cell is the serous type of cells. And one other thing which we can mention is the serous glands cells are cuboidal, central spherical nucleus. As you can see here, cuboidal cells for serous gland. As compared with the mucinous glands, they are having that empty look, mucoid secretion, foamy appearance, and they are columnar cells. As you can see, oval basal nucleus. The height of cell is more than the breadth of the cell. So mucoid cells are columnar, whereas the serous cells are cuboidal. Now, serous cells can also be having some characteristic features which we are going to look at in the next coming slide. Here we have taken one of the serous cells from the serous gland, and these cells, they are having polarized cytoplasm. We'll find that towards the base we are having rough endoplasmic reticulum, and that is why towards the base there is more of hematoxylin taken, dark staining, blue in color, due to that rough endoplasmic reticulum and the ribosomes, as is evident here. Near the nucleus, towards the basal side, more darker stain, but towards the apical side, towards luminal side or secretory aspect, there are some zymogen secretions. So in the apical cytoplasm, there is more of eosinophilic staining. As is observed here, these secretory vesicles towards the apex, they have zymogen granules and could be more pinkish because of more eosinophilic staining. And when it comes to the Golgi apparatus, it is usually supranuclear. And as is evident, here is the nucleus. Electron microscopic picture, this is the nucleus with the nucleolus evident here. And then near the nucleus, you have rough endoplasmic reticulum with a lot of ribosomes, which is evident here. This is all rough endoplasmic reticulum. Membrane-bound stacks of organelles with attached ribosomes involved in making some proteins, packing them into secretory vesicles, which then reach and join membrane-bound stacks of the Golgi bodies, which will then repackage it, and those secretory vesicles are then sent towards the luminal side. They'll fuse with the plasma membrane and send the secretions out. So here, this is the nucleus region, this is the region of rough endoplasmic reticulum, and here are the Golgi bodies repackaging the secretory vesicles. And those secretory vesicles, they become secretory granules like lysosomes covered by cell membrane, as are evident here. And then the membrane will be fusing with the apical cell surface, and this is the luminal side. Here, the secretions are then moving out. These are the secretory granules. This is the region of Golgi body, supranuclear in location, as we have mentioned earlier as well. And in this electron microscopic picture, you can see the mitochondria also, along with the rough endoplasmic reticulum. So there are some diagram-based questions with markers, and we have to identify various cell organelles in them. Talking about the histology of skin, skin will have the upper portion, which is epidermis, and the lower portion, which is dermis. In the epidermis, we have skin epithelium, which is having 5 layers if you have taken the specimen from thick skin like sole and palm. But if it is thin skin like the dorsum of the hand, then there are only 4 layers. The stratum lucidum is missing from the thin skin. We'll see that in a diagram. As you notice, this is a specimen taken from the thick skin like that of the palm and sole. Hence, in the skin epithelium, you have 5 layers. The very superficial layer, as represented by C, it is the stratum corneum, and it is having lot of keratin fibers, as you can see, represented by this eosinophilic appearance. Now, the second layer is this pinkish thin layer, and that is represented by L, the stratum lucidum. As we have mentioned, this stratum lucidum, the thin pinkish layer, is seen only in the thick skin, will be missing from the thin skin. Now this number 1, stratum corneum, number 2, stratum lucidum, they have dead cells. The cell organelles are compromised, and these layers are gradually getting shuttered off. Desquamation, loss of the layers keep happening from the superficial surface of the skin. Now the third layer, which is represented by the G, is the stratum granulosum, named so because it is having some granules, keratohyalin granules. granules, as the name is suggesting, will be giving us the keratin filaments. So this third layer, the stratum granulosum, having some granules, the keratohyalin granules helping us forming the keratin fibers. Actually, keratin fibers are formed by the keratinocytes, which are more evident in the fourth layer, the stratum spinosum. Now this layer, which is the thickest of all, the stratum spinosum, the fourth layer in the thick skin, is called so because it is giving us the spiny appearance, pointed appearance of the cells after the histological preparation. It happens because the cells here are getting detached during histological preparation, and they were actually having some desmosomal attachments, the spot welding. And wherever the spot welding was, those points, they become spiny, hence the name spinosum. And these desmosomes, they are compromised in pemphigus vulgaris, autoantibodies to destroy the desmosomes, and hence there will be intercellular separation in the living tissue, leading to skin blisters. And that is majorly affecting layer number 4, stratum spinosum. Then there is a single layer of cell towards the basement membrane, and it is called stratum basale, the fifth layer. So here, a single cell layer, which is usually columnar cells, the stratum basale, is also called as the stratum germinativum because that is where you have the stem cells, the germ cells, which are adding more and more cells towards the upper side, adding more layers, because as the superficial layers are being lost, you must keep on adding new layers from below. This is the skin epidermis. If you look at the dermis, there will be a superficial papillary layer of dermis, and then there's a deep reticular layer of dermis. That reticular layer is not evident here, but in the papillary layer, which is some loose areolar connective tissue, you can find there is one arrow mark showing one receptor here, which is Meissner's corpuscle. Let us magnify this diagram and look at some detail of Meissner corpuscle as well, which you find here. It is encapsulated structure with stacks of cells, and this Meissner corpuscle is one of the receptor which is in the papillary layer of the dermis. It is to detect the 2-point discrimination, tactile discrimination. So that we will remember and discuss again when we are looking at the details of the receptors. But at the moment, we have one line diagram for the layers of the epidermis, but this time we have taken it from thin skin. That means we'll not have the second layer or stratum lucidum. It will be missing from there. It's a line diagram and we can move on to that now. So here you can see the layer number 1, stratum corneum, and layer number 2 is missing. So directly comes layer number 3, which is stratum granulosum with keratohyalin granules, and then layer number 4, the stratum spinosum, and this is the layer number 5, a single cell layer, the stratum basale. So what about the first layer, stratum corneum? Lot of keratin fibers, where you can see there is some detachment of the dead cells as well. So this is the keratinized stratified squamous epithelium, where the keratinized layers are being desquamated. Second layer missing because it is thin skin. The third layer comes the granular layer. And this granular layer is having keratohyalin granules, which will be helping us to form the keratin. And the next layer is stratum spinosum, which is also called prickle cell layer. Prickle means spiny, pointed cells, as the appearance comes after histological preparations. And a lot of desmosomes are holding the cells together, which can be detached in intraepithelial lesion, which is pemphigus vulgaris, producing skin blisters, some autoimmune disorder. And the last layer, which is a single cell layer, stratum basale or stratum germinativum. Now, this stratum basale, why it is called stratum germinativum? Because of the stem cells. As you can see, these stem cells, they are supposed to divide and keep giving more and more layers from below, so that when there is loss of skin layers from the periphery, you must add on from below. In the basal layer, you will have some other cells as well. A cell which is called melanocyte, derived from the neural crest cells, will be giving us melanin to counter the ultraviolet radiations in the sunlight. And you can see it is giving multiple processes which are moving into upper layers like stratum spinosum. And in the basal layer, stratum germinativum, you have one receptor also, Merkel cell, a slowly adapting receptor for light touch sensation. Like you are holding a pencil, it can detect that light touch sensation, the Merkel cell in the basal layer. Then there is a description of Langerhans cells. These Langerhans skin cells are actually antigen presenting cells, and they're not in the basal layer. They are basically in the middle layers like the stratum spinosum or the prickle cell layer, and they belong to monocyte phagocyte series. They pick up the antigen and present it to the lymphocytes as they move on towards the lymph nodes. Now we are supposed to look at the papillary layer of dermis at the dermo-epidermal junction where we could see the Meissner corpuscle. We want to compare the Meissner corpuscle with the Merkel cell, which is in the basal layer of epidermis. We want to compare the receptors with each other now. Once again, we look at the structure of the skin, which is showing the epidermis and the dermis. Dermis is having two layers. There is a superficial papillary layer of dermis, which is quite thin as compared with the deep reticular layer of dermis, which is having a larger proportion of connective tissue. Now, talking about the receptors, we have seen in the stratum basale or stratum germinativum, there is a slowly adapting receptor, which is the Merkel receptor. So, Merkel receptor is a slowly adapting receptor to detect a light touch sensation like holding a pen in your fingers. That can be detected by the Merkel cells. As we were comparing it with the Meissner corpuscle, the Meissner corpuscle is actually at the dermo-epidermal junction in the papillary layer of dermis, the superficial layer of dermis, and this is encapsulated receptor with multiple stacks of cells for two-point discrimination, the Meissner corpuscle. Now, this Meissner corpuscle is a rapidly adapting receptor as compared with the Merkel cell. Merkel cell is slowly adapting receptor. So what is the purpose of discussing that? Actually, the blind people, when they are reading the Braille, there comes a question. Which of the two? Is it the Merkel receptor or the Meissner receptor? The most precise to detect the Braille is actually the Merkel cell. But since it is a slowly adapting receptor, it can identify the characters. Cannot help us in reading the sentences because then you are moving faster. And that is why reading the Braille, to understand the language of Braille, you cannot depend upon the Merkel. Merkel may be identifying the character but not helping us to understand the sentences. So for that purpose, the Meissner is going to help us in reading the Braille faster and making sense of the sentences. So question number 1, which is the best receptor, most precise receptor to identify the character of Braille? In that case, your answer is obviously Merkel cell. But Merkel cell is used by the new learners who have just started to learn the Braille. What about the experts who read fast and who can actually make sense of the Braille language? In that case, you have to use the rapidly adapting receptor, and that is the Meissner corpuscle. Now, the Meissner corpuscle is also helping us in two-point discrimination, tactile discrimination, and it can also help in detecting low-frequency vibrations. Then which receptor is helping us in detecting high-frequency vibration? For that, you have to come to the deep dermis, and there you have this Pacinian corpuscle. Pacinian corpuscle is detecting pressure and vibration, and the vibration also high-frequency vibrations. It is in the deep dermis, and in the deep dermis, you also have the Ruffini receptor. The Ruffini receptor is to detect the dermal stretch. When there is a stretch on the skin, that can be detected by the Ruffini receptor. It is a slowly adapting receptor, whereas the Pacinian is rapidly adapting receptor. So now we know there are two of them which are rapidly adapting and two of them which are slowly adapting. The rapidly adapting are Pacinian and the Meissner corpuscle, and the slowly adapting receptors are the Ruffini receptor and the Merkel receptor. Talking about the cell junctions, these are the connecting links between the two cells, holding the two cells together. And as we enumerate them, towards the apical surface, we are having the tight junctions. Below that, we have the zona adherens. Then still below is the macula adherens. Further down is the gap junction. And then we can talk about the cells attaching to basement membrane with the help of what is called as hemidesmosomes. So here we can notice that there is cell number 1 here, cell number 2, connected with the help of cell junctions. Here itself is cell number 1, cell number 2, interconnected by cell junctions. These arrow marks are showing the cell junctions. Then there will be some intercellular space also, where you have these cell junctions. Towards the apical surface, we can see there are some apical modifications, which are the microvilli. So as we are representing, these are the microvilli, which are some surface modifications. Now, towards the apical surface, we are having the tight junctions, and these are called tight junctions because the intercellular gap is very tight, very narrow. As you can see, this is cell number 1 and this is cell number 2, and between them, as you can notice, the cell boundaries, very narrow intercellular gap. So this is the tight junction, and it is a semi-permeable type of junction. Means it will not allow the molecules which are outside to move inside, like water molecule or some infective agents like bacteria. They cannot move between the two cells, so they are impermeable to certain molecules. Tight junctions, and they are like a belt all around, circumferential manner, so that it is connecting one cell with the surrounding cells all around. This is the tight junction, which is also called as zona occludens, and this zona occludens will be having the occludin proteins which are helping to hold the cells together. Now the next cell junction is zona adherens, and this adherens junction is having cell adhesion molecules called as calcium modulated adherens, the cadherins. So this zona adherens, where you have transmembrane proteins, the cadherins, will be connected to some of the actin filaments in a belt-like fashion, circumferential manner, again connecting one cell all around with the other cells which are in the periphery, holding them together. Now the intercellular gap at the level of zona adherens is 20 nanometer. So this comes as a question, what is the gap between the two cells? Like you see here, this is the level of zona adherens, and at the level of zona adherens, it is 20 nanometer of intercellular gap. So this gap between the two cells is 20 nanometer. That is how you are going to identify the zona adherens and differentiate it from the tight junction, because there the intercellular gap is very narrow. Now you add +5 and then you are reaching what is called as macular adherens. So at this level, you will see the intercellular gap has become +5. This is the level of desmosome. And when you are talking about the desmosomes, where you are having 20 + 5, that means 25 nanometer of intercellular gap. The same thing you can see here, 1, 2, 3. And these desmosomes, they are basically also called as macular adherens. Macula means spot. So they are in a spot-like fashion, not like a belt. They'll be holding the cells with each other, but only at certain spots. So macular adherens or desmosomes or spot welding is the other name. They can be compromised in pemphigus vulgaris, autoantibodies towards the cadherins, the cell adhesion molecule which are working even for desmosomes. And desmosomes are destroyed, the cells are separated from each other, which can result in skin blisters, pemphigus vulgaris. And if you go still below that, between the cells you have the gap junctions. These gap junctions, again the intercellular gap become very narrow, and in the electron microscopic picture you will find that the gap is again becoming narrow. So very little portion of the diagram is showing us the level of gap junction. We can make it out because the wide intercellular gap is now becoming narrow. So at the level of gap junction or the tight junction, intercellular gap is very narrow. Otherwise, zona adherens, it is 20 nanometer. At macular adherens, it is 25 nanometer. Now, what about this gap junction? How is it formed? It is having hexagonal connexons, making a tunnel between the two cells. So these are some hexagonal connexons, and they are making a tunnel between the two cells. And this tunnel, which is between the two cells, is like a metabolic coupling. There can be free exchange of molecules between the two cells, like glucose molecule, amino acid molecules, or ions like calcium ion, sodium ion, according to concentration gradient. And it is working like electrical synapse. Say for example, in cardiac muscle, one of the cardiac myocyte has received the impulse, is contracting, but simultaneously the next cell must also be contracting to have rhythmic contraction. So the ions from one cell like calcium ion can immediately quickly move towards the next cell through this tunnel. It is working like electrical synapse and which is quite fast. The purpose is the moment one cardiac myocyte is contracting, the other should be contracting simultaneously for rhythmic contraction, and it is understood. If there is a problem with the gap junction, then it can lead to cardiac arrhythmia. These kind of gap junctions are also found in the smooth muscles, working like an electrical synapse. As one smooth muscle cell is contracting, the other cell must also contract, as we see in peristalsis, wave of rhythmic contraction. And obviously, if there is a problem with gap junction, there will be problem of peristalsis, disturbed peristalsis. And then we are moving on to this half desmosome, what is called as hemidesmosome. And these hemidesmosomes, they are helping the cell attaching to the basement membrane. And in bullous pemphigoid, there are autoantibodies which are destroying the integrins of hemidesmosome, separating the cells from the basement membrane. Bullous pemphigoid is a subepithelial lesion as compared with pemphigus vulgaris, which is actually intraepithelial lesion, destruction of the desmosomes. A question comes, in the cardiac muscles, we are having the intercalated disc which is connecting the two cells with each other. There will be some cell junctions, like this is one of the intercalated discs. So at the level of intercalated disc or intercellular cell junction, which cell junction is missing? And we have to answer the tight junctions are missing. And which junctions are present? What we have here will be the adherens junction, that is zona adherens, and we'll have the macular adherens, that is desmosome, and we'll have the gap junctions also, but tight junction will be missing. Now we have mentioned gap junctions work like a tunnel between the two cells, like electrical synapse, which is allowing the movement of ions between the two cells. If one cell is contracting, it has received one impulse, and that impulse must be transmitted quickly to the next cell so that it also starts contracting for rhythmic contraction. So of course they'll have gap junctions working for electrical synapse. Number two, when these cells are contracting, there is a tendency they might try to separate from each other. To hold them together, we have what is called as adherens junction and the desmosomes as well. Now, how do you know that this is a cardiac muscle? Because cardiac muscles will show intercalated disc, like one is shown there, another one is here, which are basically at the junction of the two cells, like mentioned here, intercalated disc. They are typical feature of cardiac muscle. Number two, cardiac muscles are actually branching and anastomosing. So you can see branching and anastomosing feature of the cardiac muscles. Plus they have perinuclear halo. This is the nucleus, and around that you see empty space, perinuclear halo. Another nucleus here and perinuclear halo. Now why there is perinuclear halo? Why empty space? Because in that space you do not have the myofibrils, and that is why it appears like empty space. These myofibrils, which are there in the muscle fibers, they are also arranged in a regular pattern. That is why striations are evident. So in cardiac muscle, you can see dark band, light band, dark band, light band. Dark band, the A band, and isotropic. Light band, the I band, isotropic. In polarized light, these bands become more prominent. And this happens, the striations happens due to regular arrangement of the sarcomeres, the structural and functional unit of muscle. But we have to remember, these striations are less evident in cardiac muscle and more evident in the skeletal muscles. Let us put the skeletal muscle along with the cardiac muscle and also compare them with the smooth muscles now. So as we talk about the muscle tissue, we have to look at the skeletal muscle, the cardiac muscle, and the smooth muscle. The skeletal muscles are under the somatic nervous system. We can move the skeletal muscle at our own will. But when it comes to cardiac and the smooth muscles, they are under the autonomic nervous system. They are visceral muscles and we cannot control them at our own will. Autonomic nervous system and somatic nervous system. Moreover, the skeletal muscles during development will lose their cell membrane, so they become anatomical syncytium. Let us look at the diagrams and explain it further. As you see, this is a skeletal muscle and we are comparing it with cardiac and the smooth muscles. We already mentioned cardiac and smooth muscles are under the autonomic nervous system, whereas the skeletal muscle works under the somatic nervous system. And in addition, the skeletal muscles, the striations are quite prominent as compared with the cardiac muscle, which are having less prominent striations, the A and I band, whereas in the smooth muscles, the sarcomeres are not at all regularly arranged. That is why no striations, non-striated. This is less striated. This is prominently striated, the skeletal muscle. And as I mentioned, in the skeletal muscle during development, the cell membranes were lost, and it is kind of anatomical syncytium. The cells become quite long. So as you draw them, you'll find long cylindrical cells having multiple nuclei, and that too at the periphery. So it is an anatomical syncytium with no cellular boundaries, multinucleated cell, and these multinucleated appearance. Also, the nuclei are at the periphery. Plus, these I-band and A-bands are quite prominently placed. Comparing it with the cardiac muscle, they are functional syncytium. And what do you mean by that? See, cardiac muscles have intercalated disc, but at the level of intercalated disc, they are having a tunnel, which is the gap junction. And through the gap junction, there are free exchange of molecules. It is like a metabolic coupling, electrical synapse. So it becomes a functional syncytium, though there is a presence of cell membrane. But functionally, it is not working. The two cells are intercommunicating. Plus, the striations are less prominent due to less regular arrangement of sarcomeres. Then we mentioned there is a perinuclear halo, as evident here. And that happens because that open space, there are no myofibrils available. And the cardiac myocytes are branching and anastomosing. So you can see the cardiac muscles are like this. This is one of the cardiac myocyte, one nucleus here, and now it is communicating with another cardiac myocyte with one nucleus. And the site of junction is where we have intercalated disc, where we'll have the cell junctions we have mentioned, but at the intercalated disc, the cell junction which is missing is tight junction. They do not have tight junction. And what about the smooth muscles? Smooth muscles are actually different in the sense that they are spindle-shaped, very short cells with a single nucleus, and for the purpose of peristalsis, even they will have what is called as the tunnels or the gap junctions, working like electrical synapse, so that if one of the cardiac myocyte is receiving an impulse and is contracting for peristalsis, the next cell must contract in a wave of peristalsis. And obviously, if these tunnels between the cells, the gap junctions are compromised, there will be disturbed peristalsis. And obviously, they will also have desmosomes to hold the cells together while they are contracting individually. In connective tissue, we have discussed the various types of fibers making the connective tissue and its types, various sites where we can see different type of examples. As it goes with the classification, we'll find that the connective tissue could be loose connective tissue or dense connective tissue. Loose means you'll have less of fibers as comparatively having more of fibers in the dense connective tissue. Now, under the category of loose connective tissue, we can have loose areolar connective tissue, which can be seen in the subepithelial connective tissue, like we are talking about the superficial papillary layer of the dermis. There we see loose areolar connective tissue under the epithelium, and it can also be seen in the lamina propria. For example, in the GI tube, subepithelial connective tissue, the lamina propria, is also having loose areolar connective tissue. When we are talking about the dense connective tissue, that could be dense regular connective tissue, where you have regular arrangement of constituting fibers. For example, a tendon or a ligament. But when it comes to the dense irregular connective tissue, the fibers are not arranged in regular fashion. They are arranged in irregular fashion. For example, covering of the bone, periosteum, or covering of the cartilage, the perichondrium. Now, this is the diagram which we are going to enlarge and talk about the thick type of fibers, which are collagen fibers, as we compare with the thin fibers, which are the elastic fibers. Plus, there can be a third type of fibers, which are short and branching fibers, the reticular fibers. So let us begin with the loose areolar connective tissue now. In this loose areolar connective tissue, we have less of fibers, and the fibers we are talking about can be three. As we mentioned, it could be the collagen fibers or the elastic fibers, and then the reticular fibers. Though we understand the reticular fibers are the least common among the three. Collagen fibers, as are evident here, they are thick and comparatively longer and running in bundles. We are comparing it with the elastic fibers, which you will find are shorter in length, run singly, and they are branching as well. So as we mentioned, the elastic fibers are SSSB. The first S is they are short in length, and then they are straight in the sense that the collagen fibers are making wavy bundle of fibers, and the elastic fibers are running single, not in bundles, and they branch. So here we see the marker talking about the elastic fibers as one is seen here. And for reticular fibers, we have got another diagram. Now there came one question asking about ligamentum flavum. Which kind of fibers do you have there? Let us look at a diagram for the same. Ligamentum flavum is present in the vertebral canal where spinal cord is running, and it is one of the ligament protecting the spinal cord. It is actually connecting the lamina of the higher vertebra with the lamina of the lower vertebra. So interlaminar ligament seen from the posterior view and from the side view. Which kind of fibers does it have? 80% fibers are elastic fibers and 20% are collagen fibers. And now we want to look at a diagram for reticular fibers as well. The reticular fibers, reticular connective tissue, is having very fine thin network of fibers. In fact, reticular fibers are type 3 collagen fibers, and these type 3 collagen fibers are making a network, or you can say a framework of fibers supporting the cell types. It is providing a supporting framework. Like you will find, it could be seen in the liver or kidneys and the lymph node, but we have to be careful. Thymus, despite being a lymphoid organ, does not have reticular fibers. Then how do you support the lymphocytes in the thymus? If you do not have a supporting framework of reticular fibers, in the thymus, the framework is being formed by what is called as thymic epithelial reticular cells. So they will be forming the network in which the lymphocytes can be suspended and supported. Thymus is the organ which does not have reticular connective tissue. Comes as a question. Now let us look at the regular and irregular type of dense connective tissue. Dense means we'll have more of fibers as compared with the loose areolar connective tissue. So the fibers what we see here are the collagen fibers. When we say regular versus irregular, it is seen that in regular connective tissue there is wavy bundle of collagen fibers running parallel to each other in one particular direction, say along the force of the pull of the muscle where it is attaching to bone. So this is a regular or parallel arrangement of fibers running in one particular direction. As we mentioned, can be seen in the tendons or ligaments, even a flat tendon, what is called as aponeurosis. Now as you compare it with dense but irregular connective tissue, irregular means the collagen fibers are running not arranged parallel, not in one particular direction. As you can see, the fibers are seen running north, east, west, and south, and they do not have one particular direction. A group of collagen fibers running this way, the others are running this way, and this kind of arrangement, the dense irregular connective tissue, can be seen in the periosteum covering the bone, perichondrium covering the cartilage, or the deep reticular layer of dermis. All of these are MCQs. We have learned that the superficial papillary layer of dermis, the subepithelial connective tissue, is loose areolar connective tissue. But when it comes to the deep reticular layer of dermis, then it will be dense but irregular connective tissue. As we have observed, can be seen in periosteum and perichondrium as well. Collagen fibers are several types, but we'll be focusing upon the type 1, 2, 3, and 4, especially talking about their location. And in this table, we can find the most common of the 4 is the type 1. In fact, the fiber type in body is 90% collagen. So where exactly do we find this most common type of collagen or the type 1 collagen? There are several examples. Like you can say 80% of the fibers in the skin are type 1 collagen. Type 1 collagen is in the bone, in the tendons, in the ligaments. And when it comes to type 2 collagen, we can talk about the cartilages. But in the cartilage also the hyaline and elastic cartilage, because when we say the fibrocartilage, it is atypical cartilage. And in fact, the fibrocartilage will be having type 1 collagen, just like the bone. So it is atypical in nature. And if we remember, type 3 collagen is the reticular fibers. As we have discussed, these reticular fibers are going to form supportive protective network so that some of the cells like lymphocytes can be held together. When we were talking about the reticular connective tissue, the type 3 collagen, we mentioned the lymphoid organs, like here it is mentioned as the spleen or maybe the lymph node. But then we mentioned type 3 collagen fibers, reticular fibers are absent in thymus despite being a lymphoid organ. So it will have a supporting network of thymic epithelial reticular cells. It does not have the reticular fibers. Type 3 collagen can be found in the recent healing tissue. If there is injury and there is some healing going on, it'll have type 3 collagen fibers. But if you're talking about the old scar, then we have to change the answer to type 1 collagen. So type 1 collagen in old scar, type 3 collagen in recent healing tissue. All of these are MCQs. And coming to the type 4, the collagen is found in the basal lamina. For example, it is present in the kidney or the capsule, which can be compromised in Alport syndrome, some glomerulonephritis, and problem with the lens capsule. Problem of type 1 collagen is seen in the osteogenesis imperfecta, where we have weak brittle bones, and deficiency of collagen 2 can result in problem of cartilage like achondrogenesis, dwarfism. So we have seen the 4 type of collagen, their locations, and problems that might arise resulting from their deficiencies. Cartilage tissue is 3 types, which we can discuss in the coming diagram. The most common type of cartilage is the hyaline cartilage, whereas the least common is the elastic cartilage. We'll be looking at various places where these type of cartilages are present, but at the moment, let us just compare them with each other. Normally, we compare the hyaline cartilage with the fibrocartilage. So what will be the differences between the two? Cartilage will have chondrocytes, cartilage cells present in cave-like spaces, that is the lacuna. Now, the chondrocytes in hyaline cartilage are present in clusters. So, these are the chondrocytes present in the lacuna. They are in clusters, group of 3-4 chondrocytes together. But that is not observed in case of fibrocartilage, because there we find it is rows of chondrocytes. As you can observe, they are making lines. One chondrocyte is running after the other chondrocyte. So, why they are arranged in rows? Because in fibrocartilage, where you have more fibers and less of chondrocytes, there is alternate band of the rows of chondrocytes with the fibers, basically the collagen fibers. And as we have learned earlier, fibrocartilage is atypical cartilage, so the type of collagen in fibrocartilage is type 1 collagen. The collagen type in the hyaline and elastic cartilage is type 2 collagen, but fibrocartilage is atypical, having type 1 collagen. And this collagen fibers, as we know, they are running as long wavy bundle of fibers, the collagen fibers, and they are alternating with the rows of the chondrocytes. And these chondrocytes, they are actually running one after other, making some rows. So this is fibrocartilage. What about its comparison with the hyaline cartilage? Any other point? We find that the hyaline cartilage has the hyalos appearance, glass-like appearance. Despite the fact that it is having the collagen fibers, which we mentioned, type 2 collagen fibers, those collagen fibers are not visible to our eyes in preparation. So why there is glass-like appearance, or what you say hyalos appearance in the hyaline cartilage? Because the refractive index of the matrix is same as the refractive index of the collagen fibers. So they are merging up. It's an optical illusion, and our eyes cannot see the collagen fibers despite the fact they are present in the matrix. So giving a hyalos or glass-like appearance. Moreover, the matrix in hyaline cartilage takes more of deep or purple stain, because they have more affinity for the hematoxylin, which if you compare with the fibrocartilage, they are more eosinophilic, more pinkish fibrocartilage, more bluish purple than it is hyaline cartilage. Now, coming to the elastic cartilage. In elastic cartilage, along with the type 2 collagen fibers, we have elastic fibers. That is why the name is elastic cartilage. So, along with the chondrocytes in the lacuna, you will have these elastic fibers, for which you have to stain specifically. Now, how do you identify the elastic fibers? Elastic fibers can be identified by the fact that they are SSSB. And what is the full form of that? They are short, straight, running single, and branching fibers. So we can mention they are short, straight, not wavy like collagen fibers, run single, not in bundles like collagen fibers, and they are branching. So like this, they are seen, and they need some specific stains. So this is how we find the elastic cartilage is present. These elastic fibers will be giving elasticity to the cartilage. For example, if you're looking at the external ear, the pinna, you deform it and it comes back to its original shape because it has elastic cartilage. Now let us look at the various locations we can find these different type of cartilages. As we mentioned, the most common type of cartilage is the hyaline cartilage, and here it is shown in blue color. We have to understand that most of the bones in our body will be developing in the hyaline cartilage by the process what is called as endochondral ossification. So all this hyaline cartilage, the fetal cartilage, will change itself into bone, but not at the ends. At the ends of the bone, you will find there is articular cartilage shown in this blue color, and this articular cartilage, which is the hyaline cartilage, will always persist. So articular cartilage is the hyaline cartilage which never ossifies. But why? Because at the ends of the bones, like here the upper end of the humerus or the lower end of the humerus, at the ends of the long bones we find these are the articular cartilages. Like you see upper end of the femur or lower end of the femur, upper end of tibia or the lower end of tibia, those blue colors, they are all hyaline cartilage known as the articular cartilage. Articular cartilage, they have the property that they'll keep the ends of the bone smooth for smooth movements at the synovial joint. And since these articular cartilages are intra-articular, they have no perichondrium, and that is why the repair is very minimal. We know with age there'll be osteoarthritis, degeneration of the articular cartilage, and that articular cartilage, once it is degenerated, cannot repair itself because it does not have perichondrium. So articular cartilage has no perichondrium. Repair is compromised. You can see some blue color shown here as well. Anterior ends of the ribs. We understand that the vertebra and the ribs, they also develop in hyaline cartilage by endochondral ossification like the other bones. But not all the hyaline cartilage will be ossified. The anterior ends of the ribs, as shown in blue color, they are costal cartilage, still hyaline cartilage. Hyaline cartilage is also present in the respiratory tube, starting from the nose towards the larynx, trachea, bronchus. They are all having hyaline cartilage, but there are few exceptions. Like in the larynx, the epiglottis is not hyaline cartilage. It is in fact elastic cartilage. Otherwise, most of the time when we talk about the respiratory tube, it is mostly hyaline cartilage. Now let us talk about the fibrocartilage, which is working like a shock absorber and usually present in the joints. So this fibrocartilage, which is working like a shock absorber, where do you find it? In the joints, like you see, the knee menisci in the knee joint is fibrocartilage, working like a shock absorber between the bones like the femur bone and the tibia bone. Plus, you will see fibrocartilage in the pubis symphysis. The two hip bones, they are connected with each other, and there is a shock absorber in between, the fibrocartilage. We see the fibrocartilage also present in the midline intervertebral joint, the cartilage of the intervertebral disc, working like a shock absorber. So as we understand, the two vertebrae, they are holding on to each other with a shock absorber in between. The intervertebral disc is made up of fibrocartilage. We see fibrocartilage also in the articular disc of sternoclavicular joint, temporomandibular joint. So articular disc at these joints are also made up of fibrocartilage. Now coming to the third type, the least common type, elastic cartilage, we have to remember one mnemonic, E cube. And what is the full form of the E cube in elastic cartilage? The first E for the E cube is the external ear, the pinna. We have mentioned it earlier as well. The E external ear, the pinna, has elastic cartilage. Then we have the eustachian tube or the auditory tube having elastic cartilage. And the third E is the epiglottis, lid over the larynx. So epiglottis is elastic cartilage. We mentioned respiratory tube is mostly hyaline cartilage with few exceptions like epiglottis. Epiglottis is not hyaline. It is elastic cartilage. So it is the external ear pinna, eustachian tube, epiglottis, which are the three examples of elastic cartilage. When we talk about the lymphoid tissue, we are supposed to discuss the primary and secondary lymphoid organs, which can be seen in the next diagram. And these primary lymphoid organs are involved in lymphopoiesis, formation of lymphocytes, and they are going to have what is called as the stem cells. Though the stem cells are formed by the bone marrow, they are later transferred to the thymus as well. So stem cells will be present in two of the organs, that is the bone marrow and the thymus. And the presence of the these stem cells make them qualify as primary lymphoid organs. The secondary lymphoid organs do not have these stem cells. Now, the red bone marrow, which is forming the stem cells and which are reaching the thymus is supposed to train them as T lymphocytes. And these T lymphocytes trained in thymus provide cell-mediated immunity required for fighting with the bacteria, protozoa, etc. Whereas the stem cells which are there in the red bone marrow migrating towards the secondary lymphoid organs to mount the immune response, those stem cells are actually going to form what is called as the B lymphocytes. And these B lymphocytes, they change into plasma cells, and these plasma cells are supposed to form the antibodies, humoral immunity. So there are two types of immunity mechanism which we talk about. One is the humoral immunity, which is antibody mediated immunity, or the one which relates with the B lymphocytes, and then we have the cell mediated immunity related with the T lymphocytes, the cells which were trained in the thymus. Now, thymus and bone marrow are primary lymphoid organs. What about the secondary lymphoid organs? The secondary lymphoid organs, they will receive the B or T lymphocytes, could be the lymph nodes or the tonsils, spleen, or the malt, mucosa associated lymphocyte tissue. Like in small intestine, terminal ileum, we have got Peyer's patches. Peyer's patches are the lymphoid aggregations which are found in the mucosa of the intestine. So these are the secondary lymphoid organs. They do not have the stem cells, but they do have the B and T lymphocyte to mount the immune response. Now we are going to compare the structure of the lymph node as related with the thymus, and then we'll be looking at the spleen structure as well. Let us begin with the details of lymph node by putting one histological picture here. And we can mention that in the structure of lymph node, we have a lot of blue dots here. In fact, these blue dots are representing the lymphocytes. And why the lymphocytes are appearing like blue dots? Because these cells have a very large nucleus occupying almost all of the cytoplasm, taking a lot of hematoxylin, so it is the blue stain. There is a very thin peripheral rim of cytoplasm which is not so evident. That is why lymphocytes are appearing like blue dots everywhere. And they can be seen not only in the lymph node but also in the thymus or in the spleen or any lymphoid organ. Now, how do you identify the lymph node? In the lymph node, we have to understand that there are some afferent vessels. And these afferent vessels, afferent means incoming, these afferent vessels will bring the lymphatic fluid into the lymph node. Lymph node is like a filter of lymph, searching for the antigen and mounting immune response upon that. Now, that lymph will be collected in the subcapsular sinus, and from the subcapsular sinus, it is moving towards the cortex, towards the paracortex, and then reaches the medulla. So, let us look at the direction of the lymph flow. From the subcapsular sinus, it is going into the cortex, then into the paracortex, then towards the medulla. So this is the direction of the lymph. And from the medulla, this lymph is moving towards the hilum of the lymph node and passing out through what is called efferent lymphatic vessel. Efferent means outgoing. Let me zoom this area. You can understand the direction of the lymph flow. So there is one incoming lymphatic vessel, efferent, and there is one outgoing lymphatic vessel, efferent. And the lymph is passing through the subcapsular sinus and then the cortex in the periphery, then slightly deeper the paracortex, and from the paracortex towards the medulla. And from the medulla, which is having the medullary sinuses, it will be then passing towards the hilum of the lymph node and going out through the efferent lymphatic as is shown in this direction. Now, at the hilum, there are some blood vessels coming, and these blood vessels are reaching the level of the paracortex. Let us show the blood vessel now. Say this is the blood vessel coming and it is reaching the paracortex. In the paracortex, what we have is high endothelial venules, and there will be some leakage of lymphocytes. So, as the blood vessels are reaching the paracortex region, there are high endothelial venules, leakage of the lymphocytes. The T lymphocytes will stay in the paracortex, whereas the B lymphocytes will move still outside and reach the cortex. And the cortex will have what is called as lymphatic nodules or lymphoid follicles. So, what we see here is T lymphocytes in the paracortex, whereas the B lymphocytes are reaching in the cortex, in the lymphoid nodules. So as we zoom out, you can mention the same story again. What we notice here is this is the capsule, some collagen fibers. Under capsule, we have subcapsular sinus. In the lymph node, we can identify the structure by noting that there is a large subcapsular sinus, which is collecting the lymph, as we have mentioned, by the the afferent lymphatic vessel. Now this lymph has to move towards the medulla, but passing through cortex, paracortex, then reaching the medulla. So here is the subcapsular sinus having the lymph which is passing along with the trabecular lymphatics. Trabecula is the partition sent by the capsule from the cortex towards the medulla and reaches towards the medulla. Medulla is slightly lighter stain, will have the medullary sinuses, and from the medullary sinuses, lymph can move towards the hilum and go out through the efferent vessels. So what are you having in the cortex? In the cortex, we are having the lymphoid follicles, and in the lymphoid follicles, what we have are the B lymphocytes, as we mentioned. So this is the cortex where we will be having the lymphoid follicles, and in the lymphoid follicles, we are going to have B lymphocytes. They are going to form the plasma cells, and those plasma cells will form the antibodies for the purpose of humoral immunity. Then what is there in the paracortex? In the paracortex, we are having the T lymphocytes for cell-mediated immunity. There are no lymphoid follicles here. Lymphoid follicles are present only in the cortex with B lymphocytes, and deeper to that is paracortex with T lymphocyte for cell-mediated immunity. And later, these B lymphocytes and the plasma cells which they form, they migrate towards the medulla. So in the medulla of the lymph node, you not only have the medullary lymphoid sinuses filled with lymph, but also the B lymphocytes, the plasma cells, and some macrophages as well, making some cords. So there are some cords and lymphoid sinuses in the medulla or inner region of the lymph node. And when we are looking at the thymus, it is understood that it is multilobulated structure. So why is it having multiple lobules? Because the capsule of the thymus is sending some incomplete septations which are dividing the thymus into multiple lobules. These are called as capsule sending the trabecula, and because of these trabecula coming from the capsule, the thymus is being divided into multiple lobules. And each lobule is going to have an outer cortex which is darkly stained and inner medulla. So as we see, there is one outer cortex, inner medulla, outer cortex, inner medulla. The lightly stained deeper portion is medulla. You can see the same thing here as well, marked in this region. If we are looking at the lightly stained deeper region, it is medulla. It is more eosinophilic or pinkish, whereas in the periphery, the darker staining is the cortex. It is taking more of hematoxylin, and these are all blue dots. These blue dots are the lymphocytes, which are called as thymocytes because they are in the thymus. Now, the thymocytes which are in the periphery, in the cortex, are immature, whereas the thymocytes which are in the medulla are being matured. They are more trained. So how the training goes on? We understand that thymus is a lymphoid organ, but it does not have the type 3 collagen fibers or the reticular fibers. Then how do you make the framework in thymus? There we have thymus reticular epitheliocytes, and these thymus reticular epitheliocytes, how do you identify them? They are slightly larger as compared with the lymphocytes, and they are more pinkish. We know lymphocytes are more bluish, more hematoxylin is taken there, but thymic epithelial reticulocytes, they take more of eosin. And these cells, they have multiple processes which keep interconnecting with the next epitheliocyte so as to make one reticulum in which the lymphocytes are suspended. They are protecting the lymphocytes from the antigens which are there in the blood. So they help in making what is called as the thymus blood barrier also. And there will be gradual exposure of the lymphocytes to the antigens as maintained by these thymic epithelio-reticular cells. So they're helping in training the lymphocytes and exposing them towards the antigens. And once they are trained, you can call them as T lymphocytes, thymus-trained lymphocytes for cell-mediated immunity. Now, in the medulla, we will find that some of the thymic epithelial reticular cells, they are going to form some concentric layers and what is called as thymic Hazel's corpuscle. It is present in the deeper region or the medulla region, degenerating thymic epithelial reticulocytes. And these thymic corpuscles, the Hazel's corpuscles, they are basically concentric arrangement of the thymic reticular epithelial cells with a degenerating center which is deeply pinkish eosinophilic. So this is one of the Hazel's corpuscle. In fact, we will see multiple such corpuscles in the medullary region, as we have mentioned earlier, not only in this diagram in the medullary region but also in this schematic diagram, the Hazel's corpuscles, the unique feature of the thymus. Then we look at the details of the spleen. We are going to magnify this schematic diagram, and along with that we have to put one histological picture as well, and we can do that in the next slide. So here we have to understand, as we keep on taking the sections on the spleen, spleen is also encapsulated structure, the capsule made up of collagen fibers sending some septa inside, like we saw in the lymph node or in the thymus. It is present in the spleen also, those trabeculae, but then there is no cortex or medulla in the spleen. In the spleen you have red pulp and white pulp. There will be collection of the WBCs, basically the lymphocytes forming the lymphoid follicles, and this is what you call as white pulp. So here, as we zoom this area, you can see there are some collections of WBCs, basically the lymphocytes, the lymphatic nodules, and these lymphatic nodules, they make the white pulp. If you look at the total composition of the spleen, only 20% of the spleen is white pulp, and rest of the spleen is red pulp, which is obviously 80%. Now, why do you call it as red pulp? So this is all going to be red pulp, except for these lymphoid follicles. Everything here is red pulp. Since it is having a lot of RBCs, hence the name is red pulp. But along with the RBCs, there can be some WBCs intermixed. If you look at the ratio of RBC versus WBCs, RBCs are counted in millions, whereas WBCs are counted in thousands. Obviously, if you mix them, which will be more? The RBCs. So red pulp, though it will have RBCs plus WBCs, still will have more of RBCs, hence the name remains red pulp. But when it comes to white pulp, it is purely the lymphocytes, the WBCs. So let us look at one more schematic diagram focusing upon these lymphoid follicles, the white pulp. And as we zoom out, you can notice the white pulp is having two components, lymphatic nodule as observed here, and number two, what is called as PALS, peri-artery lymphatic sheath. So first we are going to talk about these lymphatic nodules, and we are zooming this area. This 1 and 2 is white pulp, and in that also we are focusing upon number 1, the lymphatic nodules. As we have suggested, in the white pulp will have basically the WBCs. So this is one lymphoid follicle, another one here, one is seen. We have to understand these lymphoid follicles are full of B lymphocytes. So a lot of B lymphocytes are present in the lymphoid follicle, and they are going to form the plasma cells, which will then form the antibodies, the humoral immunity. Now, if the lymphoid follicles, the sphenic nodules in the white pulp are having B lymphocytes, then where do you have the T lymphocytes? Let us locate the T lymphocytes as well now. So in the white pulp, we have number 1, the lymphatic nodules, and number 2, the PALS. You see here, peri-artery lymphoid sheath. Here you notice one artery is coming, central artery, and surrounding that artery there will be T lymphocytes. That is why they are being called as peri-artery lymphatic sheath. So what do you understand by peri-artery lymphatic sheath? It is basically T lymphocytes which are surrounding the central artery. Now this is a longitudinal section. If you take a transverse section, this artery runs in the lymphatic nodule. So this is the lymphatic nodule once again, but this time we are focusing upon the artery, and around the artery you have collection of T lymphocytes making PALS. As you can see, around this artery there will be some collection of the T lymphocytes, and these T lymphocytes, they are making the PALS around the central artery. So the point is, when you are talking about the white pulp, in the white pulp we are having two things. Number 1, the lymphoid follicles, which are B lymphocytes, but then around the central artery you have collection of some T lymphocytes also, which is being demonstrated here. So this is together the white pulp. Now demonstrate the white pulp in this histological specimen as well. So we are going to zoom here. We already have mentioned the white pulp is these splenic nodules, the lymphoid nodules. They are all made up of B lymphocytes. But then if we are talking about the white pulp, you should have T lymphocytes also. Where are the T lymphocytes? Then you have to search for an artery. And as you search for this artery, around the artery you will have the T lymphocytes making peri-artery lymphatic sheath like here. So if you look at the spleen, there will be only scattered lymphoid follicles having B lymphocytes, and in that also there will be peri-artery lymphatic sheath, the T lymphocytes. Otherwise, rest all spleen, 80% of spleen is red pulp. So what is there in the red pulp? In the red pulp, you'll find the splenic sinusoids, which are filled with RBCs. Hence the name is red pulp, though there can be some WBCs as well. In the red pulp, again, there are two things. Number one, the splenic sinusoids filled with blood. And number two, on the boundary, you have reticular connective tissue. You know, spleen is a lymphoid organ. It will have a framework of reticular fibers. So those reticular fibers, they are surrounding the splenic sinusoids and they make splenic cords of Bell-Roth. And that you can show here also. These empty spaces, what you find, they are all splenic sinusoids. And surrounding these empty spaces, you are having some reticular fibers. They are splenic cords of Bell-Roth. In a schematic diagram, you can see the two components of red pulp again. So what are the two components of the red pulp? 80% the spleen is red pulp and itself has two components. Number one, it has splenic sinusoids which are filled with the RBCs mostly, little bit of WBCs as well. So you can show here the splenic sinusoids of the red pulp are filled with the red blood cells plus some of the WBCs. And what is surrounding that? The reticular fibers. Those reticular fibers which are surrounding it are better labeled here. You see, these are the splenic cords of the Bell-Roth which are surrounding the splenic sinusoids. So these splenic cords of Bell-Roth is also contributing to red pulp. So what are the two components of red pulp? It is basically the splenic sinusoids and surrounding cords of Bell-Roth. This is red pulp. And in fact, 80% of the spleen is constituted by the red pulp only. Respiratory system. Let us look at some details of the conducting system and also the respiratory portion of the respiratory system. We understand when we are talking about the trachea, the bronchus or the larger side bronchiole, even reaching the terminal bronchiole, till that level it is the conducting portion of the respiratory system. Beyond that begins the respiratory zone. A respiratory zone begins with the respiratory bronchiole. After respiratory bronchiole, we'll have the alveolar ducts, alveolar sacs and the alveoli themselves. But the thing is, when we are in the respiratory zone, there are some alveoli attached. Even in the respiratory bronchiole, you'll find there are some alveoli opening into the respiratory bronchiole. Means respiration begins right at the level of respiratory bronchiole and continues beyond that. Now, as we are talking about the trachea or the larger size bronchi, we find that the structure is that of the body tubes. There will be one mucosa, then there is epithelium lining the mucosa, which is respiratory epithelium. Pseudo-stratified ciliated columnar epithelium with goblet cells. But as we are in the upper portion, like in the trachea, bronchus, or the larger side bronchioles, there will be some hyaline cartilage also, which provides the rigidity and keep the lumen of the tubes patent. You don't want the respiratory tube to collapse because it might hamper breathing. So to maintain the patency, we have got the hyaline cartilage. And along with that, there are some smooth muscles helping us in reducing the lumen. Like there can be bronchoconstriction to reduce the lumen, but then there can be also bronchodilation under autonomic nervous system. Now, this is changing gradually as we are moving distally towards the alveolar level. We find that the height of the cells will be reducing. First we have columnar epithelium, then it will be gradually becoming cuboidal epithelium, and then it becomes the simple squamous epithelium. In the alveolus, 95% of the alveolar surface area is lined by type 1 pneumocyte, simple squamous epithelium. So columnar cells are reducing in height and gradually becoming simple squamous epithelium, which is good because when you have a thin cell, there is easy respiratory gas exchange possible. Then we will also notice that the cartilage, hyaline cartilage, is gradually reducing. You see these blue structures, they are becoming less and less at the level of bronchioles, and as the bronchiole size is becoming about 1 millimeter, very small lumen, at that time there will be no hyaline cartilage. So you can see at the level of smaller bronchiole, this hyaline cartilage is gone. It is actually replaced by the smooth muscles. No more hyaline cartilage. And we'll also notice when the lumen is becoming very small, like 1 millimeter, you cannot have goblet cells, otherwise there can be mucus plug. So at this level, even the goblet cells are missing. Actually, these goblet cells are being replaced by what is called as club cell or the Clara cells, which can help us in giving the surfactant. So these Clara cells are starting at the level of terminal bronchiole itself and are also found in the respiratory bronchiole. This was the boundary line, so it is prior to this boundary line, terminal bronchiole also having the Clara cell or club cells, and it is also beyond the boundary line. Respiratory bronchiole will also have the Clara cells. Clara cells are non-ciliated and they give surfactant lipoproteins, though most of the surfactant is actually produced by type 2 pneumocytes at the level of alveolus. And we also have to understand type 2 pneumocytes, they cover only 5% of the alveolar surface area, and they're not simple squamous. They are actually simple cuboidal cells giving surfactant. One of the clinical situation in the premature babies, if delivery occurs before the age of 28 weeks, these babies might suffer from respiratory distress syndrome because the surfactant is very minimal and there will be collapse of the alveoli. Surfactant is surface active agent to reduce the surface tension of the alveoli. If you don't have surfactant, the surface tension is very high, the alveoli will collapse. Respiratory distress syndrome due to deficiency of surfactant, due to deficiency of type 2 pneumocytes in the newborn, especially premature newborn babies. Anyhow, what all is reducing as you move proximal to distal towards the alveoli? Number one, the cartilage is reducing, and number two, the goblet cells are reducing, and number three, the height of the cell is reducing, and number four, even the cilia are reducing. So as you will find, cilia is seen in the trachea bronchi at the level of bronchioles also. Even the terminal bronchioles will have ciliated columnar epithelium, which is changing into ciliated cuboidal cells, and these ciliated cuboidal cells may also be seen in the respiratory bronchiole, but then you find that cilia are missing in the respiratory bronchiole distally, and even the cuboidal cells are distally becoming simple squamous epithelium. This is the case of respiratory bronchiole. So what should I answer if somebody is asking epithelium of respiratory bronchiole? You should answer it as ciliated cuboidal cells. And what about the terminal bronchiole? Ciliated columnar cells with Clara cells. Respiratory bronchiole with Clara cells giving surfactant. Now let us look at one diagram where we can talk about the bronchus, and then we can look at one diagram where we can talk about the alveolar level. But the first diagram is that of the bronchus, one histological picture. The trachea and bronchus, they appear the same with minor differences. So now we are focusing upon one histological picture of bronchus. And here is the lumen of the bronchus, which is lined by the epithelium, respiratory epithelium, which is pseudo-stratified ciliated columnar epithelium with some goblet cells. These empty-looking cells with foamy appearance is the goblet cells. This is seen in trachea, bronchus, and larger bronchioles as well. Now, under the epithelium, we are having some smooth muscle cells, and these smooth muscle cells help us in bronchoconstriction and bronchodilation. Then we'll find deeper to that will be some plates of hyaline cartilage, and this hyaline cartilage, which has hyalos appearance of the matrix and some clusters of the chondrocytes. Hyaline cartilage is to maintain the patency of the lumen of trachea as well as the bronchus. This diagram belongs to bronchus. Actually, trachea can be identified by the fact that trachea will have C-shaped cartilage ring. But here you don't see the C-shaped cartilage. It is actually one plate of hyaline cartilage, another plate of hyaline cartilage, interrupted plates of hyaline cartilage. This cannot be trachea. So it is slightly distal, the bronchus level. Plus you have seromucinous glands here. When there is a case of asthma, hyper-responsive tracheobronchial tree, parasympathetic system is is more powerful, more of acetylcholine, that time the secretion will become more and they might be blocking the lumen of the trachea and bronchus, resulting in difficult breathing, resulting in rhonchi. Plus, more of acetylcholine due to parasympathetic hyperactivity, the smooth muscles also undergo contraction and reduce the lumen of the tracheobronchial tree. So as the lumen is narrowed and it is filled with a lot of secretion, of course there will be difficulty breathing. That is what we call as asthma. Now in this diagram, a picture of lung alveoli also has come, and we can magnify this portion to discuss type 1 and type 2 pneumocyte now. As you magnify it, the type 1 alveolar cells are simple squamous cells, flat cell with flat nucleus and covering 95% of the alveoli. As you can see, these are the flat cells with flat nucleus, type 1 pneumocytes, and they cover 95% of the alveoli. Only 5% surface area is covered by these cuboidal cells. Central spherical nucleus, type 2 pneumocyte, and they are giving the surfactant, surface active agent, which will reduce the surface tension of the alveolus, preventing collapse of the alveolus. Along with that, type 2 pneumocyte, like you can see, it is having a central spherical nucleus, very few in number, lining the alveolar area. The third type of cell, alveolar macrophages, are also seen. Large cells with large nuclei and with some ingested foreign particles. These are the alveolar macrophages, also called as the dust cells. They pick up the antigen using their pseudopodia and even mount the immune response. In the wall of the alveoli, we'll see some capillary endothelium as well for respiratory gas exchange. As you see, these are some of the RBCs which can be seen in the capillary endothelium. The O2 in the alveolar lumen will be moving towards the RBCs, and from the blood vessels, the CO2 will be moving towards the alveolar lumen. Respiratory gas exchange through a thin cell type 1 pneumocyte, simple squamous epithelium. Gastrointestinal system. Let us start with the description of the esophagus in the beginning. And as is understood, the esophagus is a long tube lined by the non-keratinized stratified squamous epithelium, which begins in the oral cavity and continues down the esophagus. Then there will be a squamo-columnar junction because the stomach and the intestine themselves are lined by columnar epithelium. And wherever this squamo-columnar junction is located, there is a chance of metaplasia. This metaplasia can turn into dysplasia and cancer, usually happening at the lower end of the esophagus, and it is the adenocarcinoma of esophagus. Before we start talking about that, we have to understand esophagus in the beginning. We'll have the skeletal muscle fibers in the upper 1/3. In the middle 1/3 of esophagus, we have skeletal plus smooth muscles, but in the lower 1/3 of esophagus, we are having only smooth muscles. Beyond that, even the stomach and intestine, they'll have only smooth muscles. And esophagus, one specific feature is there. It'll have glands in the submucosa. Normally glands are in the mucosa, but two of the structures in the GIT, one is the esophagus and second is the proximal duodenum. They are having submucosal glands, and because these are some exceptions, they become the question. So the point is, normally glands are seen in the mucosa, not in the submucosa. But if you find glands in the submucosa, either it could be the esophagus or it will be the proximal duodenum. We'll be talking about this once again. Now, coming back to this problem we were talking about. This is a case of hiatus hernia, and because of the hiatus hernia, there is gastroesophageal reflux disease. Some acid from the stomach is moving into the lower esophagus, and because of that acid, which normally is non-keratinized stratified squamous epithelium, is being replaced by the columnar epithelium called as intestinal metaplasia, the Barrett's esophagus. So in a case of GERD, you can have a Barrett's esophagus, and in that case, the squamo-columnar junction, which is represented by a zigzag line, the Z line, that Z line, the squamo-columnar junction, which normally should be at the gastroesophageal junction, is moving upwards. That means the lower part of the esophagus epithelium epithelium is being replaced by columnar metaplasia, intestinal metaplasia, columnar epithelium with goblet cell. And proximal movement of this Z line is dangerous because now if the stratified squamous epithelium is being replaced by columnar epithelium, that metaplasia is prone to dysplasia, is prone to cancer, adenocarcinoma, lower esophagus. Let us look at this squamocolumnar junction, which normally should be near the gastroesophageal junction. So here you see this is the zigzag line we were talking about, the Z line, which is squamocolumnar junction. And the squamocolumnar junction can be seen here as well. As we understand that the whitish mucosa of the esophagus is lined by stratified squamous epithelium. You see multilayered epithelium here, whereas the lower part of the esophagus towards the stomach, a little portion about 2 centimeters of the lower esophagus is lined by columnar epithelium. So this is the squamocolumnar junction or the Z line. And whenever there is a case of gastroesophageal reflux disease, as said, moving into lower esophagus, this Z line may be progressing upward and replacing this stratified squamous epithelium, the whitish mucosa, with this columnar epithelium, the pinkish mucosa. There will be some pinkish patches moving upwards, and that is a problem. That is the Barrett's esophagus. This is a metaplasia, prone to dysplasia, prone to cancer. Looking at the stomach, in gross anatomy, stomach will be having 4 parts, which we can show in the next diagram. As we understand, the esophagus will be opening into the stomach, bringing the food inside, and then the stomach will be pushing the food towards the duodenum. This is the proximal duodenum. So, the first part of the stomach towards the esophagus is known as the cardia, and the last part of the stomach towards the duodenum is known as the pylorus. Whereas most of the stomach is having body, and this elevated portion in the body is called as the fundus. So in gross anatomy, it will have 4 parts. That is number 1, the cardia, and number 2, the fundus, number 3, the body, and number 4, the pylorus. But histologically, the stomach has only 3 parts. Since this fundus and the body, number 2 and number 3, they are together considered as the fundic region. So in histology, there is a fundic region which corresponds to the body as well as fundus of gross anatomy. Now, it is the fundic region which is going to produce the acid, and acid is being produced by the parietal cells. As you will mention, the parietal cells, which are also called as the oxyntic cells, they are giving us not only hydrochloric acid but also the intrinsic factor for vitamin B12 absorption. Now, if a question comes, parietal cells are predominantly located at which region? Your answer should be parietal cells are predominantly present in the fundic region of stomach. But in that also, if you have to choose between fundus or the body, you should preferably choose the fundus region over the body portion. Now, this acid which is produced in the fundic region will have a tendency to move towards the lower esophagus, leading to some ulceration in the lower esophagus. Or this acid can be moving towards the proximal duodenum and producing duodenal ulceration. So how do we prevent that? You must neutralize that acid, and this acid which need utilization before it is moving into the esophagus is the region that cardia portion of the stomach will have more of mucus secreting cells, so as to neutralize the acid. And the same thing you can find that even in the pyloric region there will be more of mucus producing cells, so as to neutralize the acid and preventing the ulceration in the duodenum. So we can mention the cardia region and the pyloric region. They are having more of mucus secreting cells, so as they can neutralize the acid and prevent ulceration in the lower esophagus or proximal duodenum. And obviously then in the cardia region of the stomach and pylorus region of the stomach, you will have less of parietal cells, less of acid production. So more of mucus producing cells and less of the acid producing cells, that is the parietal cells. And one other thing, if you remember, we have discussed in the esophagus and the proximal duodenum, we we have glands in the submucosa, which is one unique feature for esophagus and duodenum. Normally, the glands are in the mucosa. Let us look at the gastric glands now, which are there in the mucosa of the stomach. In the stomach, there are gastric pits, and those gastric pits are continuous with the gastric gland. Let us look at one diagram where we have shown both the gastric pit and the gastric glands as well. And as you see, this is the gastric pit, which is then continuous with the gastric gland. The junction between the gastric gland and the gastric pit is called as the isthmus. So as you see, isthmus means the junction, and it is a junction between the upper portion gastric pit and the lower portion gastric gland. Gastric gland will have certain parts, like it will be beginning the neck portion, then it will have body portion, and then it will have a base. Base sometimes is also called as the fundus. So fundus of the gastric gland. Now, once we have divided the gastric gland into upper portion, the neck portion, then there's a body portion, and then there's a base, the fundus portion, it is also to be understood that in the gastric gland, upper half there are 2 cells to be remembered, and lower half 2 cells to be remembered. So in the upper half of the gastric gland, which 2 cells we need to remember? As you can see, they are named as the parietal cell and the mucus cells. Actually, full name will be mucus neck cells. And as the name is suggesting, they will be producing the mucus. Whereas the parietal cells, as we mentioned, will be giving hydrochloric acid and also the intrinsic factor for vitamin B12 absorption. Now, there are 2 cells in the upper half of gastric gland. What about the 2 cells in the lower half of the gastric gland? They are the chief cells and the G cell, the neuroendocrine cells. The chief cells are giving us pepsinogen for the digestion purpose, whereas the neuroendocrine cell, as the name is suggesting, is giving endocrine secretions, hormones like the gastrin hormone. So G cell for gastrin, endocrine means hormone. Now these 2 cells are towards the lower half of the gastric gland, means towards the base or fundus of the gastric gland. So towards the base, towards the fundus, you have 2 cells, the neuroendocrine cell and the chief cell. Chief cell is blue in color. It takes more of hematoxylin. Basophilic, blue in color. BB, blue basophilic, because it is taking more of hematoxylin. Whereas the parietal cell, PP, parietal cell is pink in color because it takes more of eosin. So you can write the chief cell, which is towards the base, is basophilic or blue in color because more of hematoxylin, and PP, parietal cell, is pink in color because it takes more of eosin. One more thing, you can see that the parietal cell is cuboidal cell. Height is equal to breadth with central spherical nucleus in the upper half of the gland, and in the lower half of the gland, the chief cell is actually columnar cell. Height is more than the breadth, and there's an oval basal nucleus. Columnar cell, which is basophilic, and parietal cell, which is cuboidal and eosinophilic. Then comes a description of 5th cell, which is the stem cell. And if there is a description of stem cell which is for the repair, say for example there is gastritis, and gastritis will cause destruction of these cells, so you have to replace them. The repair can be done by the stem cell. This stem cell is located at the isthmus region. So when we are searching for the stem cell, it is at the isthmus region, which is junction of the gastric pit and the gastric gland. Though the stem cells can be found at the base also, but the first answer for stem cell should be isthmus. The location and the function is repair. It can multiply and replace any of these cells when they were damaged by gastritis. Now we are looking at one histological picture for the gastric gland, and as you can see, this is simple columnar epithelium lining the stomach, and there will be some gastric pit, and this is the gastric gland. As we have shown, upper half of gastric gland will have 2 type of cells and lower half will have the other 2 type of cells. In the upper half of the gastric gland, we have the parietal cell, which is pink in color, and the lower half of the gastric gland, we have the chief cell, which is blue in color, since it takes more of hematoxylin. So as you can see here, the upper part of the gastric gland, you see the pink color, parietal cell, PP. Parietal cell is pink in color. As you notice, comparatively, it is more eosinophilic, more pinkish. As you are comparing with chief cell, which is more bluish, purplish, the basic stain, the hematoxylin, that is why it is more blue in color, and it is towards the base. Moreover, we have discussed the chief cell is going to give us enzymes like pepsin enzyme. It has other names also. Chief cell, peptic cell, zymogen cell, because it is having zymogen granules which will be having those pepsin enzyme. And the parietal cell is also called as oxyntic cell. It was giving us hydrochloric acid and intrinsic factor. It is also giving fried egg appearance. A cuboidal cell with central spherical nucleus. Pinkish cell in the upper half of the gastric gland. These are all mostly the parietal cells evident here. Moving on to the region of the small intestine, to increase the surface area of absorption, small intestine will have three features. It will be having some mucosal and some mucosal folds which are called as plicae circulares. And one more modification which is finger-like processes in the mucosa, the villi. And then in the villi, the enterocytes, that is the intestinal cells, will have microvilli, which is again helping to increase the surface area of absorption. Now, it is to be understood that these modifications are present only in the small intestine. They are missing from the stomach, esophagus, or from the large intestine. It is present only in small intestine. Small intestine itself has three parts. That is the duodenum, jejunum, and the ileum. So, as we are talking about these three modifications, it is observed they are missing from the proximal duodenum, will be maximum in the region of the jejunum, and then reduced in number, almost missing in the terminal ileum. So it means even in the small intestine, if you look at the proximal portion or the distal portion, these three modifications are missing. So if a question comes, the maximum number of villi, microvilli, plicae circulares are located where? Your answer should be the jejunum. Here the villi are leaf shape, tall leaf shape. And when you are talking about the ileum, the villi become short and blunt, and gradually they will be missing in the terminal portion. Now, to identify the duodenum, especially proximal duodenum, as we have mentioned earlier, there are glands in the submucosa, and those glands in the submucosa, they have a specific name also, Brunner's gland. So, duodenum can be identified by submucosal glands, Brunner gland. Normally, glands are in the mucosa, that we have to remember. This is one unique finding in the proximal duodenum. And then one unique finding in the terminal ileum. There are Peyer's patches. And what is this Peyer's patches in the terminal ileum? It is basically the lymphoid aggregations. We have learnt earlier, MALT, mucosa-associated lymphocyte tissue. So there is so much of lymphocyte tissue, especially towards the terminal ileum. There are a lot of aggregation, Peyer's patches. If you see Peyer's patches, it is ileum. If you see Brunner's gland, it is the duodenum. And if you see none of that, then it is jejunum. Plus jejunum, we have mentioned, it has maximum number of the plicae circulares, villi, microvilli, and those villi are tall and leaf shape. These are all MCQs we should be careful about. So as we see, this diagram is representing the duodenum, number 1, jejunum, number 2, and ileum, number 3, the three parts of small intestine. And this duodenum, jejunum, or ileum, since it is part of the gut tube has 4 histological layers. It begins with number 1, mucosa, and under the mucosa we have number 2, submucosa, and then under the submucosa, outside submucosa we have number 3, the muscular externa, smooth muscles, and number 4, it is the peritoneal serosa. So as you can see, towards the lumen we have the mucosa, exterior to that is some connective tissue, submucosa, then inner circular outer luxural layer of smooth muscle, muscular externa, and then connective tissue with peritoneum, simple squamous epithelium, the serosa. Then what about the modification? This is plicae circulaire, mucosal fold along with some submucosa as well to increase surface area of absorption. Plus you see these finger-like processes on the epithelium, the villi. If you are still magnifying it, then you can see microvilli as well. But these plicae circulaires, villi, microvilli are maximum in the jejunum. That is what we have mentioned. One more thing we said, in the jejunum, the villi are leaf-shaped, tall, compared with the ileum. In the ileum, villi are shorter and blunt. And now, we mentioned when we are looking at the duodenum, it has one unique feature. Glands in the submucosa, submucosal glands. And these glands are having a name also. We were calling them as the Brunner's gland. Now, Brunner's glands are present only in the proximal duodenum, absent from jejunum or ileum. Ileum, you'll find one typical feature, collection of the lymphoid tissue in the lamina propria, mucosa-associated lymphocyte tissue. And this mark is present in the lamina propria of the mucosa called as the Peyer's patches, especially in the terminal ileum. So if you see Peyer's patches in lamina propria, subepithelial connective tissue, then it is the ileum. And if the Peyer's patches and the Brunner's gland are missing, then it is jejunum. Jejunum has no Brunner's gland or no Peyer's patches, though it has maximum number of plicae circularis, villi and microvilli. Now let us move on to the details of Brunner's gland in the next diagram, proximal duodenum. So as you notice, the stomach will push the food into the duodenum. This is first part of duodenum, second part of duodenum, third and fourth part of duodenum. It will then continue with the jejunum. But here in the proximal duodenum, we will see that there is presence of glands in the submucosa, the Brunner's gland, along with the glands which are there in the mucosa, which we are calling as intestinal glands or crypts of Leverkuhn. So there are some intestinal glands which are the crypts of Leverkuhn, but along with those intestinal glands, we have glands in the submucosa, also the Brunner's gland. And you can see there are some villi, finger-like projections to increase the surface area of absorption. And if you are still magnifying, there will be microvilli forming striated border. So in the small intestine, we also have microvilli producing the striated border to increase the surface area of absorption. Now these Brunner's gland, what is their function? You see, the Brunner's gland secretion is alkaline. And why do you need alkaline secretion in the proximal duodenum? Because the stomach is sending acidic chyme towards the duodenum. And that acidic chyme in the duodenum can produce duodenal ulcer. You don't want duodenal ulcer. So the secretion must be alkaline so that it can neutralize the acidic chyme, reducing the chances of ulceration. That is the function of the Brunner's gland, giving alkaline secretion and then neutralizing the acid. Plus, it will give a hormone. The Brunner's gland is giving a hormone which is called urogastron. And urogastron, which is also called human epithelial growth factor, the other name, is performing two functions, again, to reduce the chances of duodenal ulceration. So, what is urogastron doing? So, as the chances of duodenal ulceration is reduced, number one, the Brunner's gland, when they give urogastron will reduce the activity of parietal cell in the stomach. You know, parietal cell produce acid. If you reduce the activity of parietal cell, you are reducing acid. And if you are reducing acid, you are reducing the chance of acid entering into the duodenum, hence reducing the chances of duodenal ulcer formation. And number two, if ulcer has already formed, urogastron is epithelial growth factor. It can increase the mitotic activity and heal the ulcer faster. So first of all, it is preventing the ulcer formation by reducing acidity of parietal cell. And number two, even if ulcer is there, it will increase the mitotic activity and heal the ulcer faster. That is the function of urogastron secreted by the Brunner's cell glands in the submucosa of proximal duodenum. Now we are going to magnify the crypts of Leverkuhn or the intestinal glands and want to discuss which type of cells do you find in these glands. Number one, it will be the enterocyte, columnar cells with microvilli for absorption of the nutrients. And number two, goblet cell to secrete the mucus. And number three, let us focus upon the diagram, which is a magnified picture, then we can discuss. So here is one of the intestinal gland, the lower portion, and this intestinal gland of the small intestine. We can also call it as the crypt of Leverkuhn. And as we were telling, the cell types are number one, the enterocytes. These are columnar cells with microvilli to increase the surface area of absorption. Now, number one is enterocyte. Number two, you can see this empty looking cell full of mucus. That is the goblet cell. So this is the goblet cell. Number three, the neuroendocrine cell, which will give us endocrine hormones like cholecystokinin. These cells are short length and their apex is never going to reach the luminal side. Then number four, you can talk about the Paneth cell. Paneth cells are present in small intestine, mostly near the base of the intestinal gland. Paneth cells are never found towards the upper surface or towards the villi. They are always towards the base surface of the gland, and they are identified by apical eosinophilia. The apex is pink in color. And why there is apical eosinophilia? Why the apex is pink? Because of lot of lysozymes. They stain with eosin, and those lysozymes are giving us cytokines like tissue necrotic factor alpha. So basically, the Paneth cells are providing us immunity by giving us some cytokines to kill excess of the bacteria or the protozoa, maintain intestinal flora. So these are some of the cells which we find in the small intestinal epithelium, basically the glands. The large intestine is also involved in absorption, and basically the absorption of the water and electrolytes. And as we have mentioned earlier, the large intestine will not have the modifications called as plicae circularis, villi or microvilli. They are absent here. Now, as more and more of water is being absorbed, the fecal matter becomes dry, and you have to lubricate it with more and more of mucus so that finally it can be expelled out from the large intestine. So obviously, as you move distal, you need more of goblet cells because they will give more of mucus, which will then cover the fecal matter and help in its expulsion, that is the defecation. So where do you expect largest number of goblet cells? Towards the terminal portion, basically towards the rectum. Now we are going to look at the junction of the rectum and anal canal in the next diagram. And as you notice, this is the rectum, which is continuous with the anal canal. That is where we have anorectal junction. Now, if this is the anorectal junction, we have mentioned that rectum is going to have quite large number of goblet cells so as to lubricate the fecal matter and easy expulsion. Now, the anal canal is having three zones. Number 1, colorectal zone. Number 2, transitional zone. Number 3, the squamous zone. And we are going to draw them ourselves also in a schematic diagram. So, this is the anal canal having the three zones. Number 1, the colorectal zone. As the color code is showing, it is here. Now, when you say colorectal zone, which is proximal portion of the anal canal, it is having the same feature as the rectum. So, it will be lined by simple columnar epithelium. You can put number 1 in the diagram here. This is number 1, colorectal zone, having the columnar epithelium, a tall cell where height is more than the breadth, and oval basal nucleus, the colorectal zone. Now, let us come to number 2, the transitional zone. The pinkish color, as you can see, you can mark it here as well. So as we show it here, transitional zone, this is the pinkish area. Area number 2, we are calling it as anal transitional zone. And why it is called as anal transitional zone? Because in this zone number 2, anal transition zone, there is a transitional epithelium, not the transitional epithelium of the renal tube, that is the urothelium. This is not urothelium. Here the transitional epithelium means the columnar epithelium was actually changing into cuboidal, and cuboidal was changing into squamous epithelium. So you can show in the anal transition zone there is some transition going on. We see there is columnar cell which is changing into cuboidal cell and then the squamous cell. And how do you know the three types? Because if the height is more than the breadth, oval basal nucleus, columnar. If height is equal to breadth, central spherical nucleus, cuboidal. If it is a flat cell with flat nucleus, squamous. So columnar becoming cuboidal, becoming squamous, some transition going on. This is anal transition zone, zone number 2. And then comes the zone number 3, the squamous zone. As the name is suggesting, it'll have squamous cells, actually stratified squamous epithelium. So as you show, zone number 3, the squamous zone, the yellow color, as you can notice, this is the squamous zone. And in our diagram, we can show this is number 3, the squamous zone. And the epithelium here is stratified squamous epithelium. Now, we have to talk about one line. You see, there are some glands in the anal transition zone and their lower parts is producing one teeth-shaped line, dentate line. So, dentate line is the boundary line between zone number 2 and zone number 3. That is the anal transition zone and squamous zone. So, let us mark the dentate line in our diagram. It is actually a teeth-shaped line, zigzag line. So, this teeth-shaped line, zigzag line is the dentate line. It is also called as pectinate line. And this dentate line or pectinate line is the the junction of anal transition zone and the squamous zone. Then there is a description of one more line. It is called as the white line of Hilton or anocutaneous line as shown here. Now, this anocutaneous line or the white line of Hilton can be shown here. This is the Hilton's line and it is a mucocutaneous junction because beyond this there is true skin. That means it will be dry, whereas the upper portion is mucosa, so it will be wet. In the anal canal, we are having anal mucosa, which is wet surface. But then there is a mucocutaneous line, and that is a white line of Hilton. Below that, there will be true skin with hair, and it is dry surface. So it is obvious that this true skin or the mucocutaneous junction below that, if there is dryness, the stratified squamous epithelium must have become keratinized. So here, true skin means it is stratified squamous epithelium but with keratin. And because it is dry, you will have sweat gland and sebaceous glands also. So a question comes, do you have sweat and sebaceous gland in the squamous zone? You say no. In the squamous zone, because it is a wet surface, we don't have keratin. Keratin begins only below the white line of Hilton. And due to keratin, there is dryness. And to tackle with dryness, you must lubricate it. You must moisturize it. So sebaceous and sweat gland are present, but only below white line of Hilton. It is keratinized stratified squamous epithelium. But above that, this zone number 3, it was non-keratinized stratified squamous epithelium. To discuss the histological features of the liver, let us have a look over the liver diagrams, where we have taken a section of the liver and we are magnifying it, and further magnified view is available here. It is to be understood that there will be some DAV structures, that is duct, artery and vein at the periphery, and as we understand, the duct is bile duct, the artery is the hepatic artery, and the vein is the portal vein. The liver will have some hepatic sinusoids which are receiving the blood from the hepatic artery portal vein, which is running towards the central vein. So the blood which comes from these blood vessels will be poured into the liver sinusoids, and these liver sinusoids are draining the blood towards the central vein. From the central vein, there are some hepatocytes which are polygonal large cells radiating from the central vein towards the periphery and are separated by the liver sinusoids, as are evident here. Let us magnify this diagram a bit. So these are the hepatic cells, the hepatocytes, radiating away from the central vein towards the portal triad, separated by some hepatic sinusoids which are draining towards the central vein. Then we have to understand liver has dual blood circulation. 80% of the blood is brought by the portal vein, whereas 20% is brought by the hepatic artery. As you can see, the size of the lumen is displaying more blood through the portal vein, less blood through the hepatic artery. Now the portal vein is bringing the nutrients which have been absorbed from the stomach and intestine, and those nutrients are being poured into liver sinusoids. Then the hepatocytes will be taking those nutrients and metabolizing them, maybe carbohydrate, fat or protein, and the breakdown products will be poured into the liver sinusoids, which then will push it towards the central vein. From the central vein, the blood is moving towards the hepatic vein and the inferior vena cava. Inferior vena cava will take it towards the heart, then the heart will push it into systemic circulation and the nutrients can reach throughout the body. The heart is pushing the oxygen into the systemic circulation also, and that oxygen is carried by hepatic artery. Now hepatic artery brings the oxygen, pour it into the liver sinusoids. From there, the hepatocytes can take the oxygen. Now, these hepatocytes will be sending back the CO2 into liver sinusoids along with some excretory products of theirs. And the CO2 and excretory products are again passing through liver sinusoids into the central vein. From the central vein into hepatic vein, into inferior vena cava and the heart. The heart will push this blood into lung for oxygenation now and towards the kidney for the excretion of the metabolic products sent by the hepatocytes. Now, these hepatocytes are also making the bile, and they are sending the bile towards the bile duct here. As we said, there is DAV structure. The duct is bile duct, A is the hepatic artery, and V is the vein. This bile will be now reaching the second part of the duodenum, poured there for emulsification and digestion of the fat. Along with DAV structures, there will be some lymphatics also, which will carry the lymph of the liver. So where is lymph coming from? The lymph is actually present in this space of D shape, perisinusoidal space of D shape. There we have some interstitial fluid being collected as the lymph, and that can be drained towards the lymphatic vessels at the portal triad region. Now, as we magnify this area further, we can discuss some more details of space of Desaix. And as we are doing so, you will notice the space of Desaix, perisinusoidal space of Desaix, is having hepatic stellate cell of Ito. And this cell of Ito is for absorption of the fat, storage of the fat, and also vitamin A storage. Kupffer cell belongs to monocyte phagocyte series. It is a phagocytic cell working for the immunity purpose. Now, we are going to talk more about this area, and we have a separate diagram for that. So here we see the sinusoid, which is lined by the endothelial cells, but these endothelial cells are fenestrated. As we can see, these fenestrations are there in the wall. Some windows are being created. And in this blood, which is inside the sinusoid, we are having the Kupffer cell, the phagocytic cell, sending pseudopodia through the windows into the space which is called as perisinusoidal space of Desaix. In this perisinusoidal space of Desaix, we are also seeing the microvilli of the hepatocytes. So these are the hepatocytes sending microvilli which are present in the perisinusoidal space of Desaix. As you can see, this is one of the hepatocyte here. And then we can also observe that in this perisinusoidal space of Desaix, this is the hepatic stellate cell of the Ito. This cell is for the storage of the fat and also the storage of vitamin A, and this cell has a feature of repair also. If there is some tissue injury in the liver, it can repair the liver, forming the fibers. Like we understand, liver has a framework of reticular fibers. We understand reticular fibers are type 3 collagen fibers forming the framework of several tissues and organs. But sometimes if there is too much of fibers produced by this cell, pathologically it may result in liver cirrhosis, that is the fibrosis of the liver. The hepatic stellate cell, which was helping us in normal conditions, may become a reason for the pathology called as the liver cirrhosis. And this is the slide of the liver showing the central vein here, and radiating from the central vein are observed some of the branching plates of the hepatocytes. And these hepatocytes, as they form the branching plates, all these dots we see are the nuclei of the hepatocytes. The hepatocytes are polygonal epithelial cells appearing more of pinkish because take more of eosin due to large number of mitochondria in them. And as we can see at the periphery, there are the DAV structures in the portal triad at the corners. So what is this D? D is the duct, the bile duct. Actually, it is the terminal branch of bile duct. So it is known as the ductule. And what is the A? A is the artery, actually the terminal branch of the hepatic artery. And then you can also show the V. The V is the vein, portal vein, terminal branches of the portal vein. And this is the vessel which is bringing 80% of the blood into the liver, carrying the nutrients from the stomach and the intestine, and the portal vein will be then sending the blood towards the central vein, passing through the liver sinusoids with the fenestrated endothelial cells, and those liver sinusoids are separating the plates of hepatocytes. So as you tell, the blood from the portal vein is running towards the central vein through the sinusoids, the blood is also sent by the hepatic artery branches towards the sinusoids carrying the oxygen. And then these hepatocytes, they are going to form the bile, and that bile is coming to the bile ductule here. Plus we have told there are perisinusoidal space of Desaix having the interstitial fluid forming the lymph, and that lymph is being poured into this lymphatic vessel. So as we see in the portal triad region, there is a lymphatic vessel as well. Now anatomically there is some indistinct arrangement of the classic lobule of the liver, which is hexagonal in appearance. So as we can see, a classic lobule is centered on the central vein, and at the periphery there are 6 corners. Like you see, this is the shape of the hexagonal structure, which will be more evident in the next diagram, the classic hepatic lobule. So in this diagram you can discuss the classic hepatic lobule, which is evident anatomically, will be discussed here. Or we can have another classification, which is portal lobule, the triangular structure, or one functional lobule, which is diamond shape, the hepatic acinus. But as we are talking about the classic lobule, which is hexagonal, we find that it is based upon the vascular drainage towards the central vein from the 6 corners made with the drainage of the portal vein or the hepatic artery. So, in comparison to the hexagonal structure, if we are talking about the portal lobule, then it is triangular and it is based upon biliary drainage towards the portal triad. So, here at the 3 corners of the triangle will be central vein and the hepatocytes draining towards the biliary duct at the portal triad. This is triangular. This is called as the portal lobule. Then there is a portal acinus, which is diamond shape, and we have magnified it here. In the long axis of the portal acinus, we'll find that there are 2 central veins at periphery, and in the short axis there are the terminal branches of the DAV structure. Now, in the lever acinus, as we are telling, we can magnify the diagram. The portal triads are sending some branches along the short axis, like the terminal branches of the hepatic artery, which will be carrying the oxygen. So, these terminal branches, as they pour the oxygen in the lever sinusoids, some zones are being created. It is understood that in the zone 1, there will be more of oxygen as compared with zone 2, which is more in the periphery, and the least oxygenation will be in the zone number 3, which is towards the central vein. Similarly, if we are talking about the terminal branches of the portal vein and detoxification, because liver has the function of detoxification done by the liver hepatocytes, the hepatocytes which are in this zone number 1 will be more exposed to the toxins as compared with zone number 2, which is still in the periphery, and zone number 3, the hepatocytes will be exposed to least of the toxins. So this is the metabolic zonation as we discussed in this diamond shape portal acinus, and this is the functional unit of liver and is the most well accepted when we talk about the liver units, the liver acinus, or what is also called as the portal acinus. Talking about the renal system, let us first have a look over some of the embryological aspect of developing kidney. Embryologically, kidney will have 2 portions. There will be one excretory system which is having nephron derived from the metanephric blastema, and once the urine is formed by the nephron, there is a collecting system to collect the urine, and that is derived from what is called as ureteric bud. The ureteric bud will form ureter, the renal pelvis, major minor calyx, and the collecting ducts also. We'll be talking about collecting system of urine later, but first we have to focus upon the excretory unit, the one which form urine, the nephron. Nephron is the structural and functional unit of the kidney. Now the kidney itself will have 2 portions. There will be outer renal cortex and inner medulla. In the medulla we have the renal medullary pyramids, and urine will be coming at the tip of the medullary pyramids. Once the urine is formed by the nephron, then it can reach the collecting system, that is the collecting duct, the major minor calyx, renal pelvis, and the ureter, having a different embryological origin. Now coming back to the nephron portion, in the nephron portion we have bunch of capillaries forming the renal glomerulus, and there is ultrafiltrate of blood, which is the urine coming into Bowman's capsule, part of the nephron. And from the Bowman's capsule, it will be the urine passing through the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, and then coming to the collecting duct. When we are talking about the renal glomerulus, that is the capillary endothelium or the Bowman's capsule or the loop of Henle, they are having simple squamous epithelium, flat cell with flat nucleus. But when it comes to the PCT and DCT, there will be simple cuboidal epithelium with microvilli. And when it comes to the collecting duct, there will be simple columnar epithelium. Now, when we are talking about the PCT, it is more longer as compared with DCT, which is shorter in length. The PCT and DCT, both of them are cuboidal cells, height equal to breadth, centrospherical nucleus, but in the PCT, there are more of microvilli and more of mitochondria. Hence, the staining of the PCT is more darkly eosinophilic compared with the DCT. And since the microvilli are numerous in the PCT, they are making brush border and the lumen get crowded. Is having less of microvilli, so it is not brush border and the lumen is not crowded. In PCT, the lumen is star-shaped. In the DCT, it is circular or oval. And when we look at the collecting duct, there will be columnar epithelium, and few of these cells will have short microvilli as well. Now coming to the function, we understand that the proximal convoluted tubule, which is having a lot of mitochondria, is actively involved in sodium and chlorine absorption. And as comparing it with the DCT is actually involved in sodium absorption, but that is aldosterone based. There is one renin angiotensin-aldosterone system, which is working at the level of the DCT and some of the DCT cells. When they are in the proximity of the afferent arteriole, will have densely packed cuboidal cells called as macula densa. That macula densa is detecting the sodium chloride levels in the blood and can help us in blood pressure regulation. And when it comes to the loop of Henle, which is lined by simple squamous epithelium mostly, it will have a descending limb and ascending limb. The descending limb is permeable to water, so it is losing a lot of water outside, and towards the deeper medulla, the urine gets highly concentrated inside the loop of Henle. Whereas the ascending limb is not permeable to water, so there is a countercurrent multiplier mechanism which will make sure that there is hypertonicity of urine towards the deeper medulla. As we said, the kidney has outer portion cortex and inner portion medulla. In the medulla, we are having the loop of Henle and the collecting duct, whereas in the cortex, we are having the glomerulus, the Bowman's capsule, the PCT, and the DCT. Now, once we have discussed the excretory portion and the front portion, we can talk about the collecting system, which will then collect the urine. There are 5 derivatives of the ureteric bud embryologically, which will be forming the collecting duct, minor calyx, major calyx, then urine moving to renal pelvis and the ureter. All the 5 of these structures will be lined by transitional epithelium, beginning at the tip of the collecting duct. We have to remember, most of the collecting duct is lined by columnar epithelium, but the tip of collecting duct begins the transitional epithelium, which is then lining the minor major calyx, renal pelvis, ureter. Then also it will be reaching the urinary bladder and the proximal urethra, all lined by transitional epithelium. Let us look at those details in the next diagram then. It is to be understood that the ureter, the urinary bladder, the renal tube, like any other tube, will be having the constituent layers. Like towards the lumen, there will be the mucosa, and then there is one muscular layer, the smooth muscles, and there could be peritoneal serosa or just connective tissue adventitia. Now, when it comes to the ureter or urinary bladder, the mucosal epithelium is transitional epithelium, the stretchable epithelium, having the umbrella-shaped cell, which can stretch to a larger extent to accommodate the urine for storage of urine. And under the transitional epithelium, there will be some subepithelial connective tissue, lamina propria. The smooth muscles are arranged in layers, which could be the longitudinal layer and the circular layer. Inner is longitudinal layer and outer is a circular layer, though there can always be presence of an additional layer. In here, the smooth muscles. In the urinary bladder, it is called as the detrusor muscle. Now, talking about the epithelium of the urethra, this is the male urethra, and we are marking it here. There will be a prostatic urethra, then membranous urethra, and then spongy urethra. So, male urethra is quite long, and we want to talk about its epithelium. Proximally, the male urethra will have the transitional epithelium, just like the urinary bladder or the ureter, whereas distally, the rule applies. Any external opening in our body is lined by stratified squamous epithelium. So, the tip of the penile urethra is stratified squamous epithelium, whereas proximal half of the prostatic urethra is transitional epithelium. What about the most of the epithelium of the male urethra? Mostly it is lined by stratified columnar epithelium. So, male urethra is mostly lined by stratified columnar epithelium except the beginning and the ending. And some books will mention it is actually pseudo-stratified columnar epithelium. So, most of the male urethra is lined by stratified columnar epithelium. If not found, you can mention pseudo-stratified columnar epithelium. So, for example, a question comes, what is the membranous urethra lined by? Membranous urethra is the content of the deep renal pouch, and obviously it will be lined by stratified columnar epithelium. What about the proximal half of prostatic urethra? Same as urinary bladder, transitional epithelium. And what about the tip of the male urethra? Stratified squamous epithelium. As you compare it with the female urethra proximally has transitional epithelium as it begins from the urinary bladder. But most of the female urethra is stratified squamous epithelium because it is short length and it is opening to the exterior. Any external opening is lined by stratified squamous epithelium. For a female urethra, the first answer should always be stratified squamous epithelium. In the male genital system, let us look at the seminiferous tubules which are present in the testes, some of the intra-testicular ducts, and also the extra-testicular ducts as well. Now, before we look at this histological section taken from the testis, let us look at the gross anatomy of the testis. As we zoom the area of the testis, we find that testis is covered by connective tissue, the tunica albuginea, and it is sending some septa inside the testis. Inside the testis, you'll find there are some seminiferous tubules where the process of spermatogenesis is taking place, formation of the sperm. Now, these sperms which are formed in the seminiferous tubules will be pushed into what is called as straight tubules, and from the straight tubules into the retate testis, and from the retate testis towards the outgoing channels, that is efferent ductules. They are all carrying the sperms towards the extra-testicular ducts, which begins with the epididymis. So the sperms will be received by the epididymis, and then it will be pushed towards the ductus deferens, and from the ductus deferens will enter the urethra as the sperms are ejaculated terminally. Now, when we talk about the epididymis, epididymis is the place where the sperms are getting progressive motility and they are also getting matured hair. Plus, they are stored in the epididymis before ejaculation. And these epididymis, the male genital tube, it is having pseudostratified epithelium with stereocilia, long cilia. In fact, they are not cilia, they are long microvilli. And the same epithelium can be seen in the ductus deferens as well. The lumen is lined by pseudostratified columnar epithelium with stereocilia. Now, we need to magnify the diagram of one seminiferous tubule, where we will see that at the periphery towards the basement membrane will have spermatogonium, and which are then dividing to form primary spermatocyte, secondary spermatocyte, which will then change into the spermatid. Towards the lumen, we have the spermatids developing into spermatozoa. And along with the germ cells, we also will have the supporting cells, sustentacular cells, which are tall columnar cells with large nucleus, lot of interdigitating processes called as the Sertoli cells. And if we look outside, lying between the seminiferous tubule, like one here, one another to the other side, so interstitial cells of Leydig. These are the cells which are giving us the testosterone hormone, like they are shown here. The interstitial cells of the Leydig, they are polygonal pink cells, appear pink because lot of lipid droplets which are involved in forming the testosterone. So interstitial cells of Leydig. But our focus is not upon these cells. Our focus should remain on the germ cells and the supporting sustentacular Sertoli cells. So let us move on to this diagram now. As we zoom out, we will notice this is one of the seminiferous tubule, which is having some connective tissue on the exterior, and then it is having a basement membrane. On the basement membrane, we are having the germ cell, and these germ cells, as we can see, starting from the basement membrane will be the spermatogonium. Then we have got the primary spermatocyte, the secondary spermatocyte, then spermatids, the spermatozoa. Spermatozoa will be towards the lumen of the seminiferous tubule. So here inside we'll have the spermatids and spermatozoa. Now the germ cells are identified by the fact that the chromatin matter is quite evident. A lot of chromosomes are evident as compared with the nucleus of the Sertoli cell, the sustentacular cell. So if we look at the nucleus of the sustentacular cell or the Sertoli cell, the other name, these nuclei are appearing different. Let us zoom this area here. So these nuclei are appearing different from the nuclei of the germ cells. And these sustentacular cells, they are tall columnar cells. As you will observe, these cells are having multiple processes which are supporting the germ cells and also providing what is called as the blood testis barrier. And to discuss that blood testis barrier, we have to move on to the next diagram. So in this diagram, as we understand, we have taken a section of the testis where we were focusing upon the seminiferous tubule, which was magnified here, and then we are still magnifying a portion of the seminiferous tubule. So as to talk about the sustentacular cells, the sustentacular Sertoli cells, they are giving physical support to the germ cells. They also provide nutrition to the germ cells, and they are creating blood testis barrier. So these are tall columnar cells. One of them is shown here, one nucleus, and another one is shown here, the other nucleus. They are having interdigitating processes which are making blood testis barrier by formation of some cell junctions. So let us demarcate the the Sertoli cell first. And as we show, the Sertoli cell, this is the basement membrane region, and from here, this cell has multiple processes going sideways and also towards the luminal side to hold the spermatids, giving support to the germ cells at various levels. And as you will notice, they are creating what is called as junctional complexes for blood-testis barrier. Let us show the sustentacular Sertoli cell on this side as well. So this is the multiple processes which they are sending towards the lateral side, and also they are pushing it towards the luminal side. And what is we wanting to tell here is these cells, their interdigitating processes are going to make some tight cell junctions and separating the germ cell from each other. Now towards the basement membrane, we are having the spermatogonium towards the periphery, and the spermatogonium, as they undergo cell division, they turn into primary spermatocyte, which is the largest of all the germ cells, and then primary spermatocyte will become secondary spermatocyte, will then become the spermatid, spermatozoa towards the luminal side. The point here is these interdigitating processes, they are allowing the germ cell to move from one level to other level, then to other level, then to other level, but then they do not allow blood cells from the periphery to move towards the interior, towards the luminal side, creating blood-testis barrier. No blood cells or any component of blood cells in the blood vessels which are in the periphery cannot move through this junctional complex to the interior. And what is the purpose of that? The purpose is we need a blood-testis barrier. These blood cells, the components, cannot be exposed to the spermatids because the process of spermatogenesis begins after puberty. So spermatids are like foreign bodies, and blood cells do not identify them as component of body. For them, they are foreign bodies, and if they are coming in contact, there'll be some autoimmune response. Some autoantibodies will be formed and they'll destroy the sperms, leading to decreased fertility or infertility altogether. So the purpose of Sertoli cell is not only to give physical support to the germ cells, giving them nutrition, but also protection of the spermatozoa from getting exposed to the components of the blood, hence preventing autoimmunity. Plus, the Sertoli cells give us hormones like inhibin, which is a negative feedback to the pituitary, so that pituitary is reducing the production of follicle-stimulating hormone. Plus, the Sertoli cells also give us Mullerian inhibiting substance during development. The Mullerian duct, which is female duct, must be suppressed. Then only the Wolffian duct will develop and male sexual characters will develop. So you must inhibit Mullerian duct. It is a Sertoli cell which is giving the hormone Mullerian inhibiting substance. In female genital system, we'll be looking at the details of the ovary, histology of ovary, then move on to the uterine tube, uterus, cervix, and the vagina. So our first focus is on the microscopic picture of the ovary. In this diagram, as we are magnifying the female genital system, we can see that this is the ovary, which will be giving us the secondary oocyte, which is later taken up by the fallopian tube. And in the ampulla, there will be fertilization of the secondary oocyte. The zygote is moving through the fallopian tube, reaches the uterus, and implantation occurs in the uterus. Then the baby will be delivered through the cervix of uterus and the vagina. Now, talking about the ovary, it is covered by some peritoneum, and that peritoneum is giving it a lining of cuboidal epithelium. Initially, this cuboidal epithelium was termed as the germinal epithelium. It is actually a misnomer because this cuboidal epithelium, though it is called as germinal epithelium, it is not so. It is in fact the peritoneal covering. And in fact, when you are talking about the oocytes, the germ cells, they are not derived from this epithelium, as the name might be suggesting. The oocytes are actually derived from the primordial germ cells, which are themselves coming from the epiblast cells. So it is the epiblast cells which will form the primordial germ cells, and then they are migrating here to the gonads and then form the oocyte. Most of the time in the ovary, what we find is primary oocyte until there is ovulation ovulation about to take place. Just before ovulation, the primary oocyte will change into secondary oocyte, and it is the secondary oocyte which is then undergoing ovulation. It is the secondary oocyte which is undergoing fertilization, and that fertilization, as we said, will be happening in the ampulla of the fallopian tube. Now, as we are talking about the primary oocyte, this primary oocyte will be first in the periphery of the ovary, which is called as the cortex. In the periphery, we have the cortex, and towards the center, we have the medulla. Into the medulla, you can find some of the neurovascular bundle is coming. The ovarian artery, ovarian vessels, and some nerves enter towards the medulla. But then towards the periphery, towards the cortex, you'll find there are some follicles. Now, what is this follicle made up of? As we magnify the diagram here, we have to understand there are 4 types of follicles normally we talk about. Number 1, it is the primordial follicle. Number 2, it is the primary follicle. Number 3, the arrow mark which is shown here is secondary follicle, and then we have got the 4th type, which is tertiary or Graafian follicle. Let us put the names now. Number 1, it is the primordial follicle. And then number 2, marker number 2, as you see, can be lined by one layer of cuboidal cells or multi-layered cuboidal cells, but they are called as primary follicle. So number 2 is primary follicle. It is number 3, which is secondary follicle. And number 4 is tertiary follicle, which is also called as the Graafian follicle. Now, all of them will be having primary oocyte, as is evident here. So this is the primary oocyte, as you can see, can also be seen here or here or here or here. So this primary oocyte, as we are talking about, is initially present in the primordial follicle. And primordial follicle, this primary oocyte, is covered by simple squamous cells. So in primordial follicle, the lining cells around the primary oocyte are simple squamous cells. And as we are moving towards the primary follicle, there they have become cuboidal cells. Now, this is the oocyte covered by cuboidal cells. And these cuboidal cells, in the beginning, are single layer, then they become multi-layered. But as the oocyte is covered by cuboidal cells, it is called primary follicle. These cuboidal cells are given a name also. They are called as granulosa cells. And these granulosa cells, as we mentioned here, they are going to contribute to a membrane around the oocyte, which is called as zona pellucida. Zona pellucida is a glycoprotein membrane which is contributed by 2 sources. Number 1, the surrounding granulosa cells, and number 2, the oocyte itself. So outside the oocyte cell membrane, you got a zona pellucida, which is a glycoprotein membrane, and it is contributed not only by the oocyte but also by the surrounding granulosa cells. These granulosa cells, they are also going to produce estrogen, but they cannot produce the estrogen until they are helped by some theca cells. And those theca cells are still outside the granulosa cells. So outside the granulosa cells, outside their basement membrane, there are some theca cells, and those theca cells will stimulate the granulosa cells to produce the estrogen. So granulosa cells have given 2 things now. Number 1, they have contributed to the zona pellucida, and number 2, with the help of theca cells, they have contributed to estrogen. The first half of the menstrual cycle will have a lot of estrogen, and that is being given by the granulosa cells. Now, these granulosa cells, as they are surrounding the oocyte, will be further divided into 2 types. You will notice that when we are talking about secondary follicle, there are some spaces developing here which are called antral spaces. That is why the secondary follicle is also called as the antral follicle, because it is having some antral spaces. So these are the antral spaces. And these antral spaces now, they are merging with each other and they become a large antral space. This time, when you have large antral space, this has become tertiary follicle or the Graafian follicle. So here you can mention there is a large antral space, and this is the tertiary follicle, where you can see the zona pellucida covering the oocyte. And around the oocyte, you have some granulosa cells forming the corona radiata. And still around the corona radiata, there are some some granulosa cells which are making this connecting stalk as well. So the granulosa cells which are making this connecting stalk and are covering the corona radiata, they are going to form what is called as cumulus oophorus. Now just before ovulation, as we can see here, the oocyte has changed into secondary oocyte and is being released from the surface of the ovary. The secondary oocyte is covered by the zona pellucida and is covered by the corona radiata, the granulosa cells, whereas the cumulus oophorus cells are left behind in the ovary. Now it is the secondary oocyte which is undergoing ovulation and fertilization. As it is fertilized, we'll find the remaining of the Graafian follicle changes into what is called as corpus luteum. Let us zoom out a little bit, and as we will notice, this is the corpus luteum, the remnant of the Graafian follicle. And if fertilization occurs, then we call it as the corpus luteum of pregnancy, and it starts giving the progesterone. The second half of the menstrual cycle will have more of progesterone. But as we know, fertilization is very rare event, and mostly the secondary oocyte is not fertilized. In that case, the corpus luteum is regressing and changes into what is called as the corpus albicans, and it will regress by the end of the menstrual cycle. So as we see, the primordial follicle and the primary follicle, they are preantral follicles, whereas the secondary follicle and the tertiary follicle, they are the antral follicles. This comes as a question. Now let us move on further to the details of the fallopian tube. And as we take the section of the fallopian tube here, we find in the genital tube there will be the various layers. Like towards the lumen, we have the mucosa, and towards the exterior there is muscular externa, and towards the outermost layer there is serosa. So there will be some connective tissue lined by the peritoneum, that is serosa. The muscular layer, as we understand, will have the smooth muscles. Those smooth muscles are helping in peristalsis of the fallopian tube, and it will be pushing the conceptus towards the uterus for implantation. The conceptus is pushed towards the uterus by the cilia also, which is lining the mucosa of the fallopian tube. In the fallopian tube, the mucosa epithelium is having two types of cells. Number one, ciliated columnar cells, as we just mentioned, and number two, nutritive cells, the PEG cells. PEG cells won't have cilia. In fact, they'll have microvilli. So this is the epithelium of the uterine tube, which is also called as the fallopian tube. And some of these cells, as you see, the black arrow, these are the cells having cilia. The cilia are important because they will be helping the conceptus to be moved towards the uterus and further implantation. Along with ciliated columnar cells, you see green markers. They are the PEG cells. And how do you identify PEG cells? As you will notice, the PEG cells are having protruded appearance towards the lumen. One shown here and one is shown here, and they have no cilia. In fact, they are having microvilli, and these PEG cells, they are going to provide nutrition to the conceptus. So this is the lining epithelium of the uterine tube. If we are talking about the ciliated cells, their nuclei are towards the base, oval basal nucleus. If the nucleus is protruding towards the luminal side, then that will be the nucleus of the PEG cells. Now this was the uterine tube. What about the epithelium of the uterus? As we talk about the uterus or the endocervix, they have the similar type of epithelium. That is the columnar epithelium. And this columnar epithelium is not having cilia. So you can find it in the endocervix as well, and it is shown here. Then what about the ectocervix, which is projecting towards the vagina? The ectocervix and the vagina will have same epithelium, which is shown here, stratified squamous epithelium. So we can mention now the ectocervix epithelium or the vaginal epithelium. It is the same epithelium which is stratified squamous epithelium, and because they are wet surfaces, this must be non-keratinized. So it is understood we'll have a squamo-columnar junction now, and this is the squamo-columnar junction. Whenever we have squamo-columnar junction, there is always a tendency of metaplasia, and metaplasia can become dysplasia. Dysplasia can become cancer. CA cervix we are talking about. So where is this squamo-columnar junction? At the junction of endocervix and ectocervix, because we know endocervix is lined by the simple columnar epithelium and ectocervix is lined by stratified squamous epithelium. But where exactly is this squamo-columnar junction in the gross anatomy? Actually, it is inside the cervix cervical canal in the beginning. So we find the squamocolumnar junction is inside the cervical canal in the beginning. But when the lady achieves the reproductive age, when she is capable of bearing babies, that time this endo-ectocervix junction or squamocolumnar junction is coming outside towards the vagina. But vagina is having the acidic environment, and that is going to change the epithelial lining. Now, what happens is when the columnar endocervix is coming out towards the vagina and is exposed to acidic environment of the vagina, there is a physiological metaplasia, means change of the type of epithelium, and endocervix, which has come outside here and was actually lined by columnar epithelium, will be changing into squamous epithelium. That means, in simple words, the columnar epithelium of the endocervix is changing into stratified squamous epithelium, and it is considered normal. It is called physiological metaplasia, and it will stay like that during the reproductive age. Once the lady is achieving menopause, what happens is this squamocolumnar junction, which was coming outside towards vagina, is going back into the cervix, but it is still lined by the stratified squamous epithelium. So everything is fine. What we understand is the squamocolumnar junction in the early age of a female is inside. In reproductive age, it comes outside towards the vagina, and after menopause, it again goes back inside. Everything stays normal, but there is always, as I mentioned earlier, a chance of dysplasia. The physiological metaplasia which we were talking about can result in dysplasia, which further can result in CA cervix. So ladies who are predisposed to cancer cervix must be monitored for transformation of metaplasia into dysplasia. In osteology, we are supposed to discuss the structure of bone at the gross level, at the histological level as well. But in the beginning, we'll be talking about the ossification as well. That is the embryological aspect. So embryologically, we will find that there are two types of ossifications. One is membranous ossification and one is endochondral ossification. As the name is suggesting, membranous ossification means there will be some mesenchyme membrane, and in that the bone will form. So membranous ossification happening mostly in the facial skeleton or the skull cap, partly in the mandible or clavicle bone. So when we are talking about the facial skeleton or the skull cap, or partly in the mandible or clavicle, it is membranous ossification. Otherwise, most of the bones, that is upper limb bones, lower limb bones, or the vertebra or the ribs, they all are developing in hyaline cartilage by the process what is called as endochondral ossification. So mostly we talk about endochondral ossification. Now here we have taken the femur bone, which is developing in endochondral ossification. What happens is during the intra-embryonic life, at 6 to 8 weeks, there will be a primary center of ossification developing. And that primary center of ossification developing in the 6 to 8 week of intrauterine life will be contributing to the shaft of the bone, which is called as the diaphysis of the bone. So this portion is all developing embryologically from the primary center of ossification. Now, what about the ends of the bones? If we are talking about the ends of the bone, maybe upper end or lower end of long bone, there will be multiple epiphyses. And these epiphyses, where are they developing from? They develop from secondary center of ossification, which will be appearing after birth of the baby, after intrauterine life. So, as we mentioned, there can be multiple epiphyses, and they'll be at the lower end or at the upper end of the bone, and they all are developing as secondary center of ossification, forming epiphyses or the ends of the bone. Now, once we have discussed the epiphyses at the ends, diaphysis at the shaft, there is a growth plate which we need to mention. In the growing children, there will be growth plate which is made up of hyaline cartilage, and as we will notice, this is the region of the growth plate at the upper end or at the lower end, and gradually as the growing children they become adults, the hyaline cartilage is replaced by bone, and that bone is called metaphysis. So one metaphysis at this end, one metaphysis at the other end. So the growth plate, which is in the growing children, will be then replaced by metaphysis upper end, and this is at the lower end, showing the developing bone. But when we are looking at the adult bone, we'll find there are two varieties. If we are looking at the peripheral bone, which is quite dense, is called as the compact bone, and then there is a spongy bone which is towards the interior. It is called spongy because like sponge, it will have some air spaces, and this spongy bone is also called as trabeculated bone. So as we are marking here in the shaft, the bone which is towards the periphery, as we have taken a section of the shaft of the bone here, this peripheral bone is compact bone. But as we are looking towards the interior where we have the bone marrow, that is where we have the spongy bone. Spongy bone is the area where we will have sponge-like lot of spaces, and that is where we will have the bone marrow. Now the same structure you can see is extending towards the periphery also, towards the end or epiphysis, but then the amount of spongy bone is more towards the end, and the compact bone is very minimal in the periphery. So as is evident here, the compact bone is reducing in thickness towards the ends of the bone, whereas the spongy bone is becoming more and more towards the end of the bone. Now spongy bone, which is also called as the trabeculated bone, is having some bony trabeculae, or you can say bony spicules, which are interdigitating plates of bone, and a lot of air spaces are there. Bone as such is having type 1 collagen, and along with the type 1 collagen, which is the fiber component, will have some cells also like the osteoblast and the osteocytes. Osteoblasts are involved in the bone formation, and osteocytes are involved in maintenance of the bone. These osteoblasts are forming the organic bone, which is called as the osteoid, and this osteoid, which is formed by the osteoblast, the organic bone, has to be calcified, as the bone needs mineralization. It is going to have calcium and phosphate ions along with hydroxyl ion. This is called as hydroxyapatite crystals, and that gives strength to the bone. Let us move on to the details of ossification now, specifically endochondral ossification. And we understand in the beginning there is a fetal cartilage, and that fetal cartilage is hyaline cartilage. So there is a cartilage model in which the bone is going to develop. In the 6 to 8 week intrauterine life, there will be one primary center of ossification coming, as we have mentioned, and this primary center of ossification is going to form the shaft of the bone, the diaphysis, as is evident here. This is the diaphysis which is going to develop from the primary center of ossification. Now, if primary center of ossification forms the diaphysis, what about the ends, the epiphysis? The ends or epiphysis will be developing from the secondary center of ossification, which appear after birth of the baby. So you can mention now, this is the secondary center of ossification appearing after birth, and this is the one which is going to form the end of the bone or the epiphysis of the bone, maybe on this side or maybe on the other side. Epiphysis come from secondary center of ossification. Now, once we have mentioned about the ends as well, it is understood that the bone has to grow in length, and for that bone will have what is called as the growth plate. Other name is epiphyseal plate. It is helping in growth in the length of the bone. So we can mark the epiphyseal plate here, and this epiphyseal plate, which is also called as the growth plate, is again made up of hyaline cartilage. But then as the growing child becomes an adult, the growth plate is also replaced by bone, and the bone which comes in its place is called as metaphysis. So in place of the growth plate, what you find in the adult bone is called as the metaphysis. So here we are having one metaphysis at one end, and there will be one metaphysis at the other end as well. Now it is understood that all the hyaline cartilage which we started with in the fetal level has become bone, but still there will be some hyaline cartilage which is evident at the end of the bone. It never calcifies, never ossifies, is called articular cartilage at this end and at the other end also. And this articular cartilage, as we talk about, is never going to ossify. It will keep the end of the bone smooth for smooth movements at the synovial joint. And this articular cartilage, since it is intra-articular inside the synovial joint, it does not have perichondrium. And because this hyaline cartilage is not having perichondrium, it cannot repair itself. Like in old age, there is osteoarthritis. Osteoarthritis is degeneration of the articular cartilage. Normally, hyaline cartilage can repair itself because of the perichondrium, but this articular cartilage, hyaline cartilage, is incapable of repair since it has no perichondrium. Now, let us look at the type of bone cells we are going to have. We also want to talk about the structural and functional unit of bone, which is called as osteon. So as we take the section of the bone, on the periphery we have the compact bone, and towards the narrow cavity we have the spongy bone or trabeculated bone, where we have bony spicules which are interconnected with a lot of air spaces. That is why the name spongy. That is where we'll have the bone marrow. Inner side is called as endosteum and outer side is called as periosteum. Now, may it be periosteum or endosteum, there we are having osteoprogenitor cells, the stem cells. So these osteoprogenitor cells, they are the stem cells which are going to give us the osteoblast, the bone forming cells. So here, as we look at the osteoprogenitor cell, the stem cell, they are going to form osteoblast. And these osteoblasts, as are evident here, they are going to form the organic matrix, which is called as the osteoid. So osteoid is the organic matrix which need to be mineralized or calcified later. But as the matrix gets calcified, having hydroxyapatite crystals, these osteoblasts, they are surrounded by the mineralized component and then they are named as the osteocytes. Osteocytes are the mature bone cells, basically coming from the osteoblast. Osteoblast is young cell, and once it is surrounded by the mineralized matrix, it is becoming older and becoming osteocyte. Osteocyte has multiple processes which are passing into the canaliculi, small canals, and through those canals it is carrying out the metabolic requirements like carrying the oxygen, CO2 or other metabolites from one osteocyte to the next osteocyte, and all these canals are basically connected with what is called as the Haversian canal. In the Haversian canal, like there are multiple Haversian canals which are running longitudinally in the bone, they are carrying neurovascular bundles, so they can bring the oxygen and take back the CO2, and there are intercommunicating channels. Those are the canaliculi. These Haversian canals are interconnected with each other in a transversely running manner, and these canals are called as the Volkmann canal. So Volkmann canals are running transversely, connecting the Haversian canal, which are running longitudinally. These osteocytes are basically supposed to maintain the bone. So as you can see, the osteocytes are older cells, they are mature bone cells and maintaining the bone. Then we have the description of osteoclast cells also. They are not bone cells, they are actually the monocyte phagocyte system. Several monocytes, they come together and then they are making one multinucleated cell, the osteoclast. So osteoclast is having multiple nuclei formed by fusion of multiple monocytes, and they are bone-eating cells, bone resorption. They help in remodeling of the bone. They are going to remove the calcium and phosphate from the bone and send it towards the blood. So bone resorption, that is a function of the osteoclast. It will help in remodeling of the bone. Now we are going to magnify one osteon here to look at the structure, talk more about the osteocyte and the Haversian canal. So as we do that, it is understood that osteon, which is structural functional unit of the bone, is having one central canal, which is also called as the Haversian canal. And in the Haversian canal, we are having the neurovascular bundle, where we'll have the artery and vein. It is understood artery will bring the oxygen and the nutrients, whereas vein will carry CO2 and the metabolic waste products. Now this central canal, how it is going to send the CO2 towards the osteocytes, which are there connected by some canaliculi and themselves sitting inside the lacuna, a cave-like space. So you see there are multiple canaliculi connecting this cave-like space, further another canaliculi set connecting another cave-like space, and in that cave-like space we have the osteocytes for bone maintenance. So basically there occurs diffusion of all these respiratory gases and the metabolic products from the central canal through the canaliculi towards the lacuna, and then canaliculi, another lacuna, and in the lacunae are sitting the osteocytes. So this is how the oxygen will reach to the periphery. CO2 will come back towards the central canal. And there are some concentric arrangement, lamellated arrangement of these osteocytes around the central canal. That is what we are calling as the osteon. If we look towards the periphery of the bone, we'll have the periosteum, outer fibrous layer, inner cellular layer, and towards the cellular layer, we have seen there will be osteoprogenitor cells forming osteoblasts. But they will be only at the periphery. As the osteoblasts are moving towards the interior, they become osteocytes, get trapped in the lacuna surrounded by the calcified matrix. And not only periosteum will have the osteoprogenitor cell, the osteoblast, but also the endosteum, which is towards the interior, towards the bone marrow cavity. There also we have the osteoprogenitor cell and the osteoblast. Osteoblasts are also present lining the central canal, the Haversian canal, forming the organic matrix. In arthrology, we are going to talk about the classification of joints and their structure, their examples. And when it comes to classification, we can tell that there are joints which are having restricted mobility, and they are called as the solid joints, whereas the joints which are having free mobility, they are freely mobile. These joints are called as the synovial joints. Then further, we can divide solid joints into two types, the fibrous variety and the cartilaginous variety, whereas the synovial joints will have several other types. Now, before we start talking about the synovial joint type, let us focus upon the solid joints, which we mentioned are two types. So these solid joints are going to be either fibrous or cartilaginous variety. They have restricted mobility. When it comes to the sutures, that is in the skull bones are articulating with each other with some fibrous tissue. Sutures are immobile in the skull region, and they are example of fibrous joint. Another example of fibrous joint is gomphosis, tooth and socket joint. And even this tooth and socket joint in the skull region is immobile. Now, when we talk about the third variety of the fibrous joint, that is the syndesmosis, we have to remember the term desmos means fiber. So this type of fibrous joint, syndesmosis, is slightly mobile joint. So first two joints are immobile, whereas the third variety is slightly mobile joint. One example which is being shown here is the middle radial joint. Another example which the examiners ask is inferior tibiofibular joint, just above the ankle joint in the lower limb. An example of syndesmosis, slightly mobile joint. Now, coming to the other variety of solid joint, the cartilaginous. Cartilaginous joint can be primary or secondary. If it is primary, we call it as synchondrosis, and if it is secondary, we call it as symphysis. Symphysis are usually in the midline. And when we are talking about synchondrosis, chondro relates to cartilage. It is primary cartilage joint, like epiphyseal-diaphyseal joint. In the growing children, there will be a growth plate, hyaline cartilage. So a piece of hyaline cartilage is connecting one bone with the other bone. Epiphyseal-diaphyseal joint is example of synchondrosis, and these joints are immobile joint. They don't move at all. Now, coming to the symphysis, secondary cartilaginous joint, they are slightly mobile joint. For example, midline intervertebral joint. Here, the vertebra themselves are covered by hyaline cartilage, and then there is a midline intervertebral disc made up of fibrocartilage. So, when one bone is connected with other bone having some hyaline cartilage lining, but sandwiched by some fibrocartilage, it is a structure of symphysis. Like one example is sacrococcygeal joint, a symphysis joint, slightly mobile joint. Another example is, as the name is suggesting, pubis symphysis. The two hip bones are lined by hyaline cartilage. Sandwiching them is a plate of fibrocartilage, slightly mobile joint, the symphysis, secondary cartilaginous joint. And now we are going to talk about the type of synovial joints. It is understood that when we are talking about skull and the vertebra joint, atlanto-occipital joint, the C1 vertebra, atlas, articulating with occipital bone of skull. Atlanto-occipital joint is the joint where we are doing the yes movement, moving the skull up and down. That yes continuous movement is happening at the atlanto-occipital joint. They are having condyles, occipital condyles. Even the atlas bone, the C1 vertebra, is having condyles. So some books will say these are condylar synovial joint, but these condyles themselves have elliptical surface, elliptical articular surface. So there's a confusion. Shall we call atlanto-occipital joint as ellipsoid or condylar? In that case, ellipsoid is always a better answer. So remember, whenever they are asking about atlanto-occipital joint, ellipsoid is a better answer than the condylar synovial joint. In the similar way, we have two more joints which are called ellipsoid: the wrist joint, radiocarpal joint, and the metacarpophalangeal joint, the knuckle joint. So here we are writing the three is of ellipsoid synovial joint. One we have already mentioned, atlanto-occipital joint, and the other two are the radiocarpal joint or the wrist joint, and the third one is metacarpophalangeal joint or the knuckle joint. So here is the wrist joint and here are the knuckle joint. Now when we are talking about the radiocarpal or wrist joint or metacarpophalangeal joint, ellipsoid joint, they also are considered as condylar synovial joint. So again comes the confusion. Shall we call them as condylar or ellipsoid? As I am mentioning, there are three joints which you have to remember as ellipsoid. And if the ellipsoid is not in the option, then your second answer in sequence, you could also call them as condylar. But ellipsoid is a better option than the condylar. They are poor example of condylar synovial joint. Then which is the good example of condylar synovial joint? The temporomandibular joint and the knee joint in the lower limb. So when we are talking about the temporomandibular joint here and the knee joint here, tibiofemoral joint, these are good example of condylar synovial joint. Femoral condyle, tibial condyle, condylar synovial joint. The condyle of mandible meeting with the temporal bone. So temporomandibular joint and knee joint are good example of condylar synovial joint. But then you will find some of the books are mentioning these two joints as modified hinge joint. What should I do now? See, that is the problem. Questions keep repeating and there can be different options available. So again, what you have to understand is whenever there is a question on temporomandibular joint, knee joint, you should answer it as condylar. But if condylar is not in the option, then you may change your answer to modified hinge. There are also examples of modified hinge joint. Then comes a question asking about saddle synovial joint. Let us memorize 4 examples of saddle synovial joint now. And what are the 4 examples of saddle synovial joint? One in the middle ear cavity called as malleus incus synovial joint. So for the saddle synovial joint, which is convexo-concave articular surface, you will find that there is one saddle synovial joint in the middle ear cavity, malleus and incus. Then 2 examples in the upper limb. One is sternoclavicular joint and one is first carpo-metacarpal joint. So as you are marking them here, number 1, the sternoclavicular joint, and then number 2, the first carpo-metacarpal joint. That is the joint in the thumb for the movement of thumb position. So as we are talking about saddle synovial joint, 2 examples in the upper limb. One is sternoclavicular joint, convexo-concave surface between sternum and the clavicle medial end. Then first carpo-metacarpal joint, which is the joint for thumb position between the base of first metacarpal and the trapezium bone. One more example for saddle synovial joint, the CC joint in the lateral longitudinal arch of the foot. Now, when you say CC joint in the lateral longitudinal arch of the foot, we can show it here. In the foot, we are having calcaneum bone and the cuboid bone, CC joint, forming lateral longitudinal arch. So as you notice, looking from the lateral side, calcaneum bone, the heel bone, and lateral bone, cuboid bone, between them, we are having the calcaneo-cuboid joint laterally forming the lateral longitudinal arch, is having convexo-concave surface, saddle synovial joint. Then what about the medial longitudinal arch, where we have the joint calcaneo-navicular joint? Which type of joint is that? Now, when we are talking about that joint, the talocalcaneo-navicular joint, this is ball and socket type of synovial joint. So, talocalcaneo-navicular joint forming medial longitudinal arch is ball and socket. The head of talus is the ball, navicular is the socket, contributing to medial longitudinal arch. Other example of ball and socket? Yes, you can take the hip joint and the shoulder joint. So, the other example examples are the hip joint and the shoulder joint. Again, they are ball and socket. And then if you are looking at the middle ear cavity, incus and stapes, the incus and stapes in the middle ear cavity, ball and socket. So for examples of ball and socket, incus and stapes, the shoulder joint, the hip joint, and the medial laryngeal arch, that is the talocalcaneonavicular joint, is ball and socket synovial joint. Now, what about the C1, C2 vertebra joint? That is between the two vertebrae, C1 and C2 vertebra. If we are talking about that, you can see the atlas vertebra has no body, and C2 vertebra, that is the axis vertebra, is having an upward projection called as the odontoid process or dens of the axis, forming a midline joint, C1, C2 joint. So C1 is the atlas vertebra, C2 is the axis vertebra, and there's a midline intervertebral joint, atlantoaxial joint. It is a pivot synovial joint, a rotatory synovial joint, and that is where we are doing the no movement. When we rotate the skull on the vertical axis, it is the C1, C2 joint helping us in no movement, and that is a rotatory joint, pivot synovial joint. Any other example of pivot synovial joint? Yes, the rotatory joint is also seen in superior and inferior radio-ulnar joint, where we are working for supination and pronation. So superior, inferior radio-ulnar joints, they are also rotatory type of joint where the radius rotates on ulna for pronation and supination. So both of them are pivot synovial joint. Now we know three examples of pivot synovial joint: atlantoaxial, superior and inferior radio-ulnar joint as well.